Yes, sir. You can start now.
So, good morning, good evening, and good afternoon, everybody who are listening different parts of the world. And today is our second day of World Congress on Adult Immunization. And probably you are all aware that from today, this World Immunization Week is started. And I'm just telling you what is the theme of this World Immunization Week. The slogan for the week's campaign is vaccinated populations, healthy communities. Vaccinated populations, healthy communities. That means apart from the pediatric vaccination, adult vaccination is of immense importance for building healthy communities. This is the slogan of World Immunization Week. And the theme is World Immunization Week 2022 theme is long life for all vaccines in the pursuit of a long life well lived. That means again, for long life, vaccines has got immense roles. So these are the theme and slogan of World Immunization Week. So we are going to start our program, and on the first session is on COVID. First session is on COVID, and we have got two eminent chairpersons with us. Professor G.C. Khilani, he is known to everybody, and presently he is working as a professor in the Department of Pulmonary Medicine and Sleep Disorders in All India Institute of Medical Sciences. His research interests includes pulmonary medicine and sleep disorders. He is serving an editorial member and reviewer of several international reputed journals. Dr. G.C. Khilani is a member of many international affiliations. He has successfully completed his administrative responsibilities. He has authored of many research articles, books related to the pulmonary medicine and sleep disorders. And we have also with us Professor Amitabh Nandi. He also needs no introduction. He had participated yesterday also. He had given the lecture on the vaccinations, the history of vaccination. He was the first director of the tropical medicine. He was the Department of Virology. He was a known. He is known to everybody. He is one of the teacher of teachers. So with this introduction, we are going to start our program. Now the floor is to Dr. Khilani and Dr. Amitabh Nandi.
So, good evening, everybody, and it's my privilege to be before you, and for which I thank Dr. Kohli. And everybody thought that vaccination is for children, and we pulmonologists were talking about adult vaccination with pneumonia and influenza, sometimes zoster, but COVID has changed the game, and COVID has shattered our life, changed the game of the way we practice medicine, changed the way we deal with the patients, changed the way we look at the clinical profile, and change the way we protect ourselves. So as far as the protection is concerned, besides infection control practices, vaccination, everybody wants to have a vaccine and go around without a mask, so that, which what United States did in May 2021, that they thought they are vaccinated. But nevertheless, the kind of mortalities we had during phase 1 and 2, when the wave 3 came, that is Omicron wave, we feel that India was protected from severe disease, if I talk of only India, because we were quite well vaccinated. And of course, Dr. Sanjay Rai is here with an encyclopedia. He also attests that we were grossly infected. About 90% of us have gotten infection. So we were protected. I'm just emphasizing the importance of COVID vaccination. Today is a very great day, I would say, where we are going to listen about from innovation to practice. We have an expert who will tell us about how it can be innovated. We have experts who will tell us how to use it. We have experts who will tell us what is the implication in community medicine. And finally, we have a clinician par excellence, Dr. Dhruv, who is going to tell. Without wasting much time, it's my pleasure to invite Dr. Shantanu Kumar Tripathi on my behalf and on my co-chair, Dr. Nandi. There's a big biodata that I can see of Dr. Shantanu Kumar, but I would actually not. I went through his biodata. He is a Director Clinical Research at the John C. Martin Center for Liver Research and Innovation, IILTS, Sonapur, Kolkata, and he is Academic Professor and Head of Pharmacology. So it's great to listen from a pharmacologist, and he has been involved with so many things which I went through, including the, he's involved with the Indian Council of Medical Research. Sir, we really want to learn from you as to how the vaccines are innovated. We are the end users, but it will be interesting to know how it comes. And if Dr. Nandi, you want to add something, please unmute yourself. Unmute yourself, please.
Nothing more to add. And I don't like to waste much time. I'm eagerly waiting to listen to Shantanu, who is a very old friend of mine. He's laughing. Okay, Shantanu, I invite you to just begin your presentation.
So let me share the screen. Is the screen visible?
No, it hasn't come.
So I would request you to share these. Yeah, it's coming now, sir.
It's not coming, no?
So you have to just put it on a slideshow.
Not in the slide mode, slideshow mode.
It's perfect now, sir.
Okay. So thank you very much. So next 15 to 20 minutes, I'll just talk on this theme of vaccine innovation and access. Whenever we talk of vaccines, in the same breath, we need to also talk about access, because vaccines are meant for vaccination, immunization. And we have in the last couple of years learned the hard way. What does it mean to avail otherwise much needed vaccine and also get ourselves vaccinated? Since yesterday, this must have been discussed several times. When we talk of vaccine innovation and access, apparently these two are two different terms, but they are so interlinked, intertwined. Vaccine innovation refers to newer vaccine products or adaptations to the existing vaccine products for improved effectiveness. And we'll discuss about the innovation. When you think of developing a newer vaccine product, what are the steps, essential steps? We'll discuss a little. And when you talk of vaccine access, that stands for increased coverage or full coverage and equity by overcoming a last mile barrier. And from our experience in the last couple of years during the pandemic, we also must admit that yes, we have done a lot, but at the same time, vaccines have done a lot, but at the same time, we cannot ignore that there was an equity issue also. Not necessarily all the people in the world were equally privileged to get themselves vaccinated when they needed them most. So there is a need of emphasis on this equity and access issue with the same vigor that is needed to develop a new vaccine. Now, it would be possibly naive to not accept and recognize the importance of vaccines. It is indeed a boon for global public health, undisputedly the most effective tool for preventing infectious diseases and improving global health. There has been remarkable progress since the day of Edward Jenner, and we have passed a long path with eradication of smallpox and the control of childhood diseases such as measles, mumps, rubella, polio. But have we done enough? And whether there are newer threats, unknown threats, and how are we prepared to face those unknown threats? The emerging infectious diseases is a great concern globally, and how with the use of vaccines as an undisputed tool, how we are going to actually embrace these emerging newer diseases. The emerging newer disease vaccine, perhaps if you go back to say 10 years or 15 years back, it has been, and to a great extent even today it may be true, that is almost like an orphan drug. There are not too many takers. Why? Because there seems to exist an inherent business challenge. Vast majority of the vaccine candidates, they are known to fail in the last phase, that is phase 3 of clinical trials, with an alleged loss of $1 billion US, and economic incentives for developing these vaccines seem inadequate. And that's where the so-called philanthropy model came up, with participation of organizations like World Health Organization, CEPI, Gavi, Gates Foundation, Wellcome Trust. So there was a need to do a little more than just leaving them to the industry, pharmaceutical industry. There was it was greatly necessary that it needs to be also pushed by non-industry partners. It's more of a public-private partnership that possibly might find the solution of this orphan drug status of EID vaccines. The mega fund approach is also another concept that is put forward, that public health commitment of different governments aligning all stakeholders with due incentives, perhaps that would hold the key. In the year 2017, it was WHO, PATH, Gates Foundation, UNICEF, and Gavi, that is the Global Alliance for Vaccines and Immunization. They launched the Vaccine Innovation Prioritization Strategy, VIPS, with the aim to drive vaccine product innovation to better meet country needs, the different country needs, and support the alliance, Gavi, the Gavi goals on immunization coverage and equity. So this is coming back again and again, that coverage and equity. So it's not enough to come up with newer vaccines unless we can ensure the coverage and equity. And this cartoon actually summarizes that what kind of principles are to be put together, and then only we can possibly expect the desired goal to be achieved. So on one hand, there is a need for innovation to address the unmet needs for newer vaccines, for confronting these unknown diseases and emerging infectious diseases, from the lab to the regulatory approval, and beyond regulatory approval also to reach up to the last mile, from approval to sustained access. And then we need to have enduring vaccine ecosystem to be developed, and then only these will be considered as the win-win partnership is the key to access. So I'm not going into details of it's otherwise a very busy slide, but it has tried to summarize the major key issues and how we can try to solve them by appropriately addressing each of them. So that's the concept that has been put forward by the International Federation of Pharmaceutical Manufacturers and Association, and they actually have created this cartoon, which is otherwise used to popularize and send the message, the right message amongst all stakeholders. Now we have some major, you can say that steps that has been taken or achievements that have been we have succeeded in the last, say, 1 decade or 1.5 decade, whereby the initial part, that discovery phase, okay, that has been made very short now. So that was a great challenge, because whether it is a vaccine or a drug, a new drug, so the drug development is a very tedious path. Okay, the initial discovery phase, and then the preclinical development phase, and then the clinical development phase, then regulatory assessment and licensing decisions, and after that, that can be made to public use. Now, so far as vaccines are concerned, the initial discovery phase has been made relatively easier now because of these developments. Number one is the faster sequencing, gene sequencing of the pathogen. In this case, if a new virus is detected, we need to isolate and identify and sequence its unique genetic code in order to get information that is needed to help design the diagnostics, vaccines, and treatments for that new infection. That's, in one word, it is called the medical countermeasures. So the major medical countermeasures are these diagnostics, vaccines, and treatments. On the other hand, non-medical countermeasures or public health countermeasures are use of mask, distancing, hand washing, etc., etc. The sequencing technology, gene sequencing technology, that has been dramatically improved in the last 2 decades. In 2003, if we, as an example, following the detection and isolation of the SARS-CoV-1, it was not called SARS-CoV-1 then, only after the SARS-CoV-2 came, we are calling it SARS-CoV-1. But that time it took several months for the gene sequencing. In 2020, on the other hand, with the SARS-CoV-2 appearing, it took just a couple of weeks for sequencing the SARS-CoV-2 virus, which helped us in synthesizing it. The synthetic generation, there also we have done a great success to investigate a virus. Earlier, it was the physical samples of the pathogens that were required, but the modern technology allows researchers to synthetically generate the viral genetic material in the lab so that they can begin to study the virus's properties earlier and faster. And all this we have experienced in the prompt development of the COVID vaccines. Okay, so it was a jet speed rather that the COVID vaccines were developed. With the advent of this artificial intelligence and machine learning, development of a new vaccine also has found a new encouragement. The AI has been used to handle the incredibly tedious process of sorting through thousands of potential compounds to find a few of them being viable and analyzing the potential approaches to vaccine development, which is far, far faster than any human team could achieve through informed trial and error. So this has really helped to hasten and to speed of the program of development. With the further improvement of technology, the artificial intelligence has the potential to improve the speed further at which the potential candidates can be reviewed. And finally, this nucleic acid vaccines, the advent of the nucleic acid vaccines, whether it's mRNA or DNA vaccines. So this is also our experience with the COVID-19, where we find they have provided the genetic, these vaccines actually provide the genetic instructions to produce an antigen that the immune system can learn to recognize and build immunity. More easily manufacture these vaccines, much more easily than the traditional vaccines. And these nucleic acid vaccines represent a historic milestone in vaccine science, given the unprecedented timeline of R&D, research and development. Now, the technology platforms really helped us. The novel technology platforms for the emerging infectious disease vaccine development are now available. Using the next generation strategies for precision on disease pathogenesis and the molecular mechanisms, it is easier for us now to develop newer vaccines. For vaccine development, we need basically the three things to succeed to control an infectious disease or prevent an infectious disease with vaccines. You need the vaccine, you need the vaccination, you need to understand the host, also host immune system, so that we can target the, we need to understand the genetic sequencing. We need to understand the molecular basis of pathogenesis of the infectious disease. That means we need to understand the pathogen better, so that we can understand the target against which the vaccine will be developed. Okay, and then we need to also understand the host and their immunity, so that we can plan our strategy how the vaccine will work. Now, all these have been made possible with these technology platforms. So these platforms are designed to address the mechanisms for infection susceptibility also in the special populations like elderly children or pregnant women or the immunocompromised. The major platform targets, they include DNA and messenger RNA, the viral vector, the viral-like peptides involved in the DNA replication, the peptide vaccines, recombinant protein vaccines, live attenuated vaccines, inactivated virus vaccines. So excepting for this live attenuated and inactivated virus, which are otherwise traditional vaccines, they all other, they are relatively new, and we have been introduced to these different new vaccine platforms in our acquaintance with the COVID vaccines. Or we have all the representatives of all the newer technologies now available for COVID. We understand them much better. Here you see, when you talk of COVID vaccine candidates by technology platform, on all these color codes are meant for the different platforms. Okay, whether it is live attenuated, inactivated, non-replicating viral vector and replicating viral vector vaccine, recombinant protein vaccine, peptide-based vaccines, virus-like particle-based vaccines, DNA and RNA vaccines, etc. And here you see that it was, of course, in April 2020. Okay, in the Nature review it came, the pipeline. So where you see that what was the position at that time, the number of projects that were going on. In April 2020, in fact, the trials, some of the trials had started. You see that only a few vaccines, they were in phase 1 trial. A good number were in preclinical or otherwise in exploratory phase, that is discovery and design phase. Most of them were at that level on 9th April 2020. So just a couple of words about the different development stages. The CEPI had classified vaccine development stages at the exploratory phase, preclinical and clinical stage. Exploratory phase is actually for developing the rationale for a vaccine-based molecular pathogenesis of the disease, to plan it and design a candidate vaccine, and then conduct the lab research, that is in vitro and ex vivo research, in order to test the idea for developing the candidate. Then comes the preclinical or non-clinical studies, where animal models are used, animal in vivo evaluation to understand how the vaccine works, or if it is likely to be safe and would work well in humans. And side by side, in parallel, while the preclinical is going on, we are also preparing for the clinical development, because in order to save time, economize on time, so that if we succeed in the preclinical development, then without wasting any further time for preparation for clinical development, we can straightway start the clinical development. Of course, there is a need for regulatory step here, that we need to submit for IND, investigational new drug application. So we need to approach the regulator, and then the regulator allowing, then only we can initiate the study in the human being for the first time. First human study can be conducted. And in clinical studies, you know that healthy volunteer studies with phase 1 for prophylactic vaccines, most of them are also in healthy volunteers. And the different phases, phase 1 is for safety and mainly for the safety and also the kinetics. Then further phases are for safety, efficacy, safety, and immunogenicity assessments. So preparing for clinical development, because ultimately we have to give the vaccine for clinical, in the clinical development phase, to human volunteers in the form of in the same form that is otherwise going to be used once it is licensed. So then you have to develop that particular vaccine candidate or the formulation. So that manufacturing technology, understanding that manufacturing technology is also important, and ensuring product quality, IND submission, and then regulatory evaluation, which is done by the regulator, of course. They will do the assessment of preclinical data, the GLP compliance, whether it has been observed, and then assessment of the product, its quality, safety, and manufacturing technology. Then only they will allow if they are satisfied with the preclinical submission. Clinical development.
15 minutes.
Yes. Clinical development would cover the 3 phases under regulatory oversight, trials conducted according to protocols, and even if the vaccine is primarily intended for children, initially in this case, the adults are actually taken into the trial for the trial, and then we can shift to the children. The phases usually progress subsequently, but sometimes the phases can also overlap. I'm skipping in the interest of the time the phases of phase 1, phase 2, phase 3 in detail, but we have just spoken about what is done in phase 1. Then in phase 2, it is basically for the efficacy. There are also control group is always there. It provides additional safety information besides the relationship between the dose and the immune response, providing initial information regarding its ability to generate the immune response, etc. And then the phase 3, which is in a few couple of thousands, that generates critical information on efficacy and safety. Efficacy means reduction of disease incidence, and also immunogenicity of the candidate vaccine, of course, in comparison with the control. The challenges, yes, developing vaccines to combat pathogens like MDR bacteria, HIV, influenza, SARS-CoV-2. There are a lot of challenges definitely are there. Rapid response strategies. Developing rapid response strategies is one challenge for controlling the emerging diseases. Predicting the type and timing of the next pandemic is another challenge, and providing affordable access to effective vaccines for every potential beneficiary is another challenge. Challenges continue beyond clinical trials also, with the mass production, the vials that is to be used, single vial or multi-dose vial, buffers, stabilizing agents, safety issues, batch-to-batch quality issues, even the adjuvant that is used, that can also have issues with that. Finally, last couple of slides, it is the access and equity is important. As we have mentioned, equity means it should be allocated across all countries based on needs and regardless of their economic status. Access to and allocation of vaccines should be based on principles grounded in the light of every human to enjoy the highest attainable standard of health without distinction of race, religion, political belief, etc. And globally, the distribution is shaped by challenging political, economic, social, diplomatic, and health-related matters. Accurate and up-to-date data and information are critical components in guiding the international community's understanding of vaccine equity and shed light on the blind spots essential for achieving the last mile on vaccine equity. Thank you. I think that is the last slide. Yes. Thank you very much.
Thank you, Dr. Shantanu.
Yes, if there is any question or comments, I can take, or else we can go to the next.
Dr. Khilnani.
A question, sir, questions will be discussed after these 4 topics.
Oh, I see. Okay. Okay, then, because I had some questions I'd like to discuss later on. Okay.
You can also post it in the chat box.
Okay. Okay. So,
Yes, Dr. Nandi, you carry on.
yes. So our second speaker, Dr. Khilani, can we invite second speaker?
Yeah, so the next speaker is Dr. Bruce Manghali from Singapore. And now we have just heard about the various phases of vaccine development and, state of the art disposition, so many things were not known to us. Now we are coming to the COVID per se, from the general vaccination development. And Dr. Bruce is going to tell us as to development of COVID vaccine. And normally Dr. Sanjay keeps on reciting that vaccine development takes minimum 10 years. And, people like Sanjay were able to give us vaccine in 1 year's time, 1.5. Were we in too much of a hurry? How the development took place, how the time became shorter than by 10 times shorter, I would say, which was actually to help the mankind. So this is what Dr. Bruce is going to tell us. Dr. Bruce, all is yours. Thank you very much.
Hello, everyone. I just wanted to thank the organizers for the kind invitation to share the AstraZeneca story with you today. This pandemic has had an incredible impact on people, on societies around the world, and I'm very proud to have been a small part of the amazing efforts by many teams around the world to develop rapid, effective solutions to prevent further loss of life. Today, I will touch mainly on the development of the AstraZeneca vaccine with a look at the platform itself and the clinical trials that led to its emergency use authorization around the world. Then I'll take a look at some of the real-world data that's emerged following its use, with a focus on data from the UK, one of the first countries to roll out this vaccine at scale, with an active research program to monitor it along the way. I also want to touch on some of the safety concerns that have been discussed with this vaccine. In order to understand how the vaccine works, let me just recap on the structure of the virus for a moment. Coronaviruses have a spike protein on their surface, which is essential for binding to the cellular receptor and ultimately for entry of the virus into the cell. This is the most critical protein for viral entry, so an obvious target for a vaccine candidate. This spike protein is also prone to point mutations, which are driving the evolution of variants of concern globally. The vaccine that AstraZeneca developed in partnership with the University of Oxford in the UK uses a non-replicating chimpanzee adenovirus vector to deliver the DNA corresponding to the coronavirus spike protein in cells. This is non-replicating for obvious reasons. We don't want the virus vector to be doing anything it's not supposed to do. The vector will still enter cells on first contact and deliver the DNA package containing the spike code, but the vector cannot replicate and go on to infect secondary cells. The DNA then enters the nucleus, where it remains as episomal DNA, ready to transcribe into messenger RNA. DNA is more stable than messenger RNA, so this allows the DNA code to last for longer than vaccines using mRNA only. Because there are human adenoviruses circulating in the population, there are concerns that this pre-exposure will have produced antibodies against the vector, which can then limit the immune response when the vaccine is given. For this reason, we've chosen a chimpanzee adenovirus, which is distinct from the human ones to minimize any vector autoimmunity. We know now that this vaccine induces strong immune responses after 1, 2, or 3 doses, so it's clear there is little or no inhibition of the immune response due to these autoantibodies. How are we able to develop this so fast? Adenovirus vectored vaccines are not new technology. These have been used for other diseases, and the platform was well established with the Oxford group. There was even experience developing a chimpanzee adenovirus vectored vaccine for another coronavirus, MERS, or the Middle East Respiratory Syndrome virus. So once it was clear what this new virus was, the team could move very fast in planning a strategy to beat it. Astonishingly, this process was very fast. It took just 104 days from when the Wuhan coronavirus sequence was available to injecting the first vaccine candidate into humans in a phase 1/2 trial. Approximately 1 month later, we started a much larger phase 2, phase 3 trial in the UK, with global trials following soon after that. Normally, this process would take years to get that far, but a unique collaboration of efforts building on many years of pandemic preparation with an acute need to develop a vaccine for this rapidly unfolding pandemic led to a truly Herculean effort from a vast number of people all over the world. In parallel, the company committed to producing doses at risk before any approvals had been obtained, and importantly, committed to making doses available to the world at no profit to the company during the pandemic. Emergency use authorization came right at the end of 2020, and the first vaccination outside of a trial was administered on the 4th of January in 2021. So how does it work? The vaccine is injected into the deltoid muscle, where the adenovirus vector enters primarily muscle cells. The DNA encoding the spike protein is released and enters the nucleus, where the DNA is then transcribed to messenger RNA that leaves the nucleus and is then translated into the spike protein, which is either processed internally or becomes membrane bound on the cell. This process triggers the innate immune responses via interactions between viral nucleic acids and pathogen recognition receptors. The expressed proteins undergo proteasomal degradation and presentation to CD8 positive T cells in association with MHC class I molecules, and may be secreted and taken up by antigen-presenting cells. Now, antigen-presenting cells may also acquire vaccine antigens as apoptotic or necrotic bodies, or may be directly activated by interaction with the viral vector. These antigen-loaded APCs migrate to draining lymph nodes, where they can prime CD8 or CD4 T cells and B cells. Phase 1 studies noted strong increases in neutralizing antibody titers and activated T cell response. It also became apparent that the first dose of vaccine induced almost all of the T cell response we would see. The second dose simply maintained those levels. So that initial priming dose is very critical in terms of shaping the overall response. Now, what did the clinical development plan look like? There were a couple of early studies that were accelerated to provide early data, which ultimately supported the initial emergency use authorization in the UK and Europe. These are shaded in yellow here. A more rigorous phase 3 trial was also started to facilitate licensure in the US. You can see that in this mulberry color. And a number of smaller trials were run in various countries to support local requirements where necessary. A pooled analysis of 4 of these early trials formed the basis of the initial vaccine dataset, including 2 trials in the UK, 1 in Brazil and 1 in South Africa. As you can see, there were some differences between these studies, and this led to some misunderstandings around the vaccine when it was first being rolled out. The dosing interval used was different across some of the studies, and there was even a mistake with the dosing in one study that led to challenges in interpreting a standard dose compared to a low dose in the backdrop of these different dosing intervals. However, on the plus side, this actually led to some very interesting insights in terms of the efficacy observed. In terms of safety, there were no concerns at all. The incidence of serious adverse events was lower in the vaccine arm than in the placebo arm. There was unfortunately 1 COVID-related death during the study, but this was also in the control arm. Now, as I mentioned, these 4 studies were pooled to determine the initial efficacy against symptomatic COVID-19. This had to be virologically confirmed by PCR with at least 1 symptom expected from COVID. There are over 17,000 participants that met the inclusion criteria for this analysis. And this is the primary analysis data. When the analysis was triggered, there were 332 cases that were COVID positive, 84 of these in the treatment arm, 248 in the control arm. And the vaccine efficacy was calculated at 66.7%. There were no hospitalizations in the treatment group, so efficacy was assumed to be 100%. Now, I mentioned earlier that the dosing intervals were different amongst these 4 trials, so that actually provided an interesting opportunity to evaluate the impact that dosing interval might have on efficacy. What they found was that the longer the interval, the greater the efficacy in all participants that had had 2 doses of what is now used in the vaccine, standard doses. The very shortest interval was only about 55% efficacious, but that increased to over 80% when they looked at those with an interval greater than 12 weeks between dose 1 and dose 2. This increase was also seen in neutralizing titers as well, providing, This increase was also seen in neutralizing titers as well, providing some mechanistic support for those observations. So when the vaccine was introduced, some countries initially adopted this longer interval, in some places out to 16 weeks between dose 1 and dose 2. However, the need to find a balance between optimal effectiveness of the vaccine and trying to ensure people receive both doses as quickly as possible led to a number of variations of that, with many countries finally opting to choose a shorter interval of 4 to 8 weeks to increase 2-dose coverage quickly. In addition to evaluating this increasing efficacy phenomenon, when the study investigators looked at all participants that received a single standard dose of the vaccine, they noticed that vaccine efficacy quickly reached about 76%, and this was maintained out to 12 weeks. There was no long-term data available at that time, but modeling suggested that protection after a single dose could be quite long-lasting. With this strong one-dose effectiveness, some countries have also been quite comfortable with leaving a longer interval between doses to ensure that as much of their population can receive at least one dose rather than just focusing on two-dose competition alone. So based on the clinical trial program, what do we know about this vaccine? It's quite well tolerated with mostly mild to moderate adverse events, as we see for most vaccines. It's highly effective, 76% against symptomatic COVID and 100% effective against severe disease. It's immunogenic with robust T-cell responses. This is important for memory responses and long-term protection. And based on the immunogenicity data we have, we expect quite long-term protection, at least 6 months. There are obviously discussions going on globally around the need for and the timing of booster doses, but I'll touch on that again later. It's simple to administer and requires no special storage or distribution requirements, making it a highly versatile vaccine designed for global distribution. Now let's look at the real-world evidence. There is an enormous amount of real-world evidence for COVID vaccines so far. Now, just to quickly summarize what we have seen is quite good effectiveness for this vaccine across all the main variants of concern from a number of different studies, 80 to 90% or higher effectiveness against severe outcomes, and 50 to 80% against mild symptomatic COVID. For most, this is quite consistent with what we've seen in the clinical trials. With Omicron, for all of the COVID vaccines, there has been a decrease in the observed effectiveness and a more rapid waning, particularly against mild infections. Importantly, though, with a booster dose, we do see quite good effectiveness against serious outcomes like hospitalization. And this is what's important, keeping people out of hospital and preventing unnecessary deaths. Comparatively, we've seen very similar effectiveness for both the AstraZeneca vaccine and the Pfizer vaccine in Europe during the Delta wave. And this was observed irrespective of the age group, as you can see here. There's also some data coming out of the UK evaluating vaccine effectiveness against Omicron specifically. Clearly, there is some waning happening with both the AstraZeneca on the left side in the purple here, and the Pfizer vaccine in the blue on the right-hand side. But you can see here the vaccine effectiveness is restored after a third dose back to levels providing significant protection. And this effectiveness is almost identical, irrespective of whether people were boosted with the AstraZeneca vaccine or an mRNA vaccine. Now, the real measure of how well these vaccines work is through effectiveness against severe outcomes. In this example, against hospitalization. Here you can see that after a booster dose, effectiveness is quite high, and again is almost identical, irrespective of which vaccine is used for the booster. Now, the last piece of data I wanted to share with you today is around the risks and perceptions of risks associated with thromboemolic events following vaccination. There have been concerns around these events, particularly with the AstraZeneca vaccine, following a number of reports in Europe and other places. While a definitive causal link has yet to be established, the EMA has added wording to our label to ensure vaccinators and patients are aware of these risks and can make informed decisions. An assessment of our own safety database information revealed a small but elevated risk of TTS after the first dose, but this was much lower after the second and now after the third dose as well. Importantly, there have been a number of studies conducted in Europe recently, some of them funded by the EMA to evaluate the relative risk of these events with different COVID vaccines and in relation to the risk in people who contract COVID disease. The key message from all of these is pretty much the same. The safety profiles for both the AstraZeneca and the Pfizer vaccines in terms of thromboembolic events is actually quite similar. But for people who contract COVID, the rate of these events is many, many times higher. So just to start to wrap up here, we know that this vaccine is very effective against the current range of variants of concern, particularly in reducing hospitalization and death. While there have been concerns around some extremely rare safety events, the overall safety profile is very good and now appears to be largely comparable with mRNA vaccines. We know that the immune response of this vaccine is quite durable, but recent data may be starting to indicate some waning protection over time. We don't know yet if a third dose booster is needed for all, but our own data indicates that a third dose will boost antibody and neutralization titers and maintain T-cell responses, ensuring continued protection. I just want to finish with a quick overview of what the real impact this vaccine has made. AstraZeneca embarked on this journey for the right reasons. We're committed to providing broad and equitable access to our COVID-19 vaccine. And to do this, we're providing the vaccine at no profit to the company during the pandemic. To date, more than 2.8 billion doses of our vaccine have been supplied to more than 170 countries worldwide. And our modeling indicates that it's helped to save 3.7 million lives. This is in addition to preventing many more millions of hospitalizations since we began this journey. This is something we at AstraZeneca are immensely proud of. Thank you for your attention today.
Thank you. Thank you very much, Dr. Bruce, for giving a perfect account of the protection imparted by AstraZeneca vaccine, which actually has been I think 85 to 90% of Indians are vaccinated with this, and there were indeed several aspersions and doubts, adverse effects, efficacy. All those things were there, and we always thought that we are having maybe we are having an inferior vaccine as compared to the Pfizer vaccine or mRNA vaccine, and but your data seem to suggest that the protection from, especially during the Delta waves, 88 to 90% protection against that seems very heartening. So we'll take the questions later. And now it's my pleasure and privilege to invite Dr. Professor Sanjay Rai, who is a professor of community medicine at the iconic All India Institute of Medical Science. And I always call Sanjay my vaccine guru, because right from the outset, I have had several meetings with him, television programs and webinars, and let me tell you, whatever he said at the outset came true over a period of time. A very innovative researcher, very candid speaker, and analytical mind he has. And if I could call him outspoken, it will not be correct, but he speaks what he thinks is right. So that's my introduction to Sanjay, and we are waiting to listen to you the impact of vaccination on dynamics of pandemic. I know it's a very difficult subject, and I think you can only handle it. Thank you.
Thank you so much, sir. Thank you very much for such a nice introduction. And so a very good evening, chairpersons, Dr. J.C. Khilani, Dr. Amitabh, all the other dignitaries, ladies and gentlemen. I have been asked to speak on impact of vaccination and dynamics of pandemic. No doubt it's a difficult task. I don't know I'll be able to do justice or not, but I will try my level best. Just I want to share the screen. Yeah. Is the screen is visible?
Yeah, it is.
Okay, sir. Thank you, sir. So I will start with this. What is the current policy on COVID-19 control? The current policy is universal vaccination. Everybody must get vaccine. Vaccine will prevent the future infection. Vaccine will prevent the future waves. That's why, and those unvaccinated, they are posing a risk to the society. So they must get vaccine. Otherwise, they are the antisocial element for the society. This is the current policy. Vaccinate all children before sending to school when parents have a lot of concern, and contact tracing is the best method to control the pandemic. Test, track, treat. This is the current policy related to pandemic, and this will decide the propagation of the pandemic. So in epidemiology, we usually teach that for any communicable disease, agent, host, and environment, they decide the propagation of the communicable disease. So it's a relevant chapter is related to, my topic is host-related immunity. I'm not going to cover the agent and environment part. We know this is RNA virus and they bound to mutate. Already more than thousands of mutations have occurred, and although their variant of concerns are only 5, the original Wuhan, then alpha, then beta, then gamma, then delta, and and now the Omicron, and Omicron, the various other lineages of the Omicron we have. So I'm not going to discuss the agent part. In question answer, we can discuss, but the host-related immunity we can discuss. So the future course of pandemic will be decided by differently level of the immunity. So level of the protection of the community. We know we have two type of acquired immunity. One is your vaccine associated, and second one is natural immunity. So I will discuss vaccine associated and strength of the vaccine associated immunity, strength of the natural immunity, and then what should be the future course of our pandemic. This is the old slide I am using since last almost 2 years. Last time, so the data I'm not going to cover. Dr. Khilani, you will recall that when first time, just after when we launched this vaccine, that time I said that definitely there will be end of this pandemic, but because this was the usual question, that is there end of this pandemic? So answer is always yes. We had several pandemic in the past also. We said goodbye to all this pandemic. So definitely there is end of this pandemic also, and through herd immunity or population immunity. That time I said that both natural infection and vaccination will provide the herd immunity, and will say goodbye to this in the future. In January, when India launched on 16th of January, the first vaccine in many countries in December 2020, they launched the vaccine. So we had a lot of hopes that time. And I said, but very soon I revised my strategy, my statement that no, we are not going to achieve herd immunity through the vaccination. We will most likely will achieve only through the natural infection. And still, I'm for natural infection, that only the natural infection is this also. Why? I'm going to discuss this. So we know there are a lot of vaccines, and the overall, the objective of all these vaccines is to reduce our severity, death, morbidity, mortality. So we have a lot of vaccines. I'm not talking about the COVID vaccines. So there are vaccines. More than 3 million lives we are saving per year. So 30 lakhs lives you are saving. We have more than 20 vaccines for different diseases. We have already eradicated your smallpox. Polio is under control. So we have eliminated from this region. There are only few endemic countries for the polio, and many communicable diseases we have already controlled. So this is the beauty of vaccination. And now we have few vaccines, not few, 37 vaccines for this COVID. The 37 vaccines have already been approved by the various regulatory authority. And so this is the current situation. 37 vaccines already approved. So around 350 vaccines are in the different phases of the development. Phase 3, phase 4, 45, and 194 are in the preclinical phase. So this is the current situation as per WHO data. This is the current world situation of the vaccination. We have already inoculated more than 65% of the world population, at least with one dose. Around 12 billion doses have been administered globally, and currently 11 million doses are administered each day. So in India, almost 100 crore, at least 100 crore population have received at least one dose. So fully vaccinated are more than the 60% in our country also. These are the various vaccines I'm not going to cover. So Covaxin is an indigenous vaccine, completely indigenous vaccine, developed here. ICMR developed, isolated the virus, and then we developed here through all the clinical trials done here. Dr. Bruce, he was talking about the AstraZeneca vaccine. They published the data in the Lancet, and efficacy was the clinical trial data of Brazil and UK data of 17,000 they published in the Lancet long back last year, somewhere October, November, December in 2020. And the efficacy was 62.2, 62.1%. We consider double-blind clinical trial data is the best evidence for any research-related evidence. So, in interventional study, first intervention is double-blind. Although this was the single-blind, but that was published in the Lancet. Sputnik. So these are the various vaccines. I'm not going to cover in India. These are the various vaccines. They are safe. We are using many are in India and effective also. Safety in terms of relative safety, in terms of if you compare with the impact of SARS coronavirus 2, that was a huge impact. Still, it's a huge impact. But these almost all vaccines we are using, they are effective and they are safe also. These are the WHO approved vaccine, 10 vaccines they have already. But see, it's not only safety, it's not in the absolute term. None of these vaccines are 100% safe. We cannot say, like any other drug, we cannot say. But it's a very rare event, like thrombocytopenia of this 1.33. If you see the incidence ratio, or if you compare with the SARS coronavirus 2, it's a 5 times lesser than the SARS coronavirus. So thrombocytopenia is 5 times more in SARS coronavirus 2 infection, that is COVID-19 patient, but almost they also have some thrombocytopenia with these vaccines. So venous thrombolism, etc. So all these are there. So in nutshell, I can say vaccines also have some serious adverse event following immunization, like messenger RNA vaccines. They are causing cardiac inflammatory disease, pericarditis, myocarditis. So there are a lot of publications. In 2 years, we have generated a lot of evidences. But these serious adverse events are very rare. Breakthrough infections are happening all over the world. Everywhere breakthrough infections are happening in fully vaccinated. UK, when even during Omicron, almost 50,000 per day they reported breakthrough infection. 50,000 per day. We have clear-cut definition of breakthrough infection. That 2 weeks after the second dose, if an infection is happening, then we'll call breakthrough. If it's a 2-dose schedule, or 15 days after the first dose, if it's a 1-dose vaccine, like Johnson & Johnson vaccine. So we have clear-cut definition of breakthrough. So breakthrough infections are happening. If you compare breakthrough infection with your natural infection or reinfection rate, breakthrough infection rates are much higher. Even we cannot compare. So example is Estonia, other country like Seychelles. Why I'm taking the examples of a small country? Seychelles is a very small country, right? This is the Seychelles and island. So if you want to study the epidemiological picture of any disease or intervention, impact of the intervention of any disease, then this type of country is always better. The interference from other part of the world will be less. If suppose if a lockdown is complete there, so the local impact. So it's although a small country, but they had this was the first country. Seychelles was the first country almost vaccinated more than 60% population in April 2021. Israel was the second country, and then they had a very big infection almost in all islands. They have almost 150 islands, so they had infections in islands. So breakthrough infection everywhere completely. They reported only 1 or 2 cases per day throughout the year before the fully this vaccination program. But after that, they reported a huge outbreak of SARS coronavirus 2 infection. So the message is the vaccine is preventing your severity and death, but not the infection. CDC in report August 16, this earlier they have presented that during July, 74% new infection in the Massachusetts, USA were breakthrough infections, where the vaccination coverage was 69%. So almost completely breakthrough. So these are not preventing infections. Then I'm talking about the natural infection. There was one study, very good study, published in the New England Journal of Medicine long back, in the beginning of, if I recall correctly, 2021. They followed up 20,000 healthcare provider, and the study design were very good. They followed for the 7-month period, 31 week, and every 2 week they tested with RT-PCR. So that design was very, really, if you want to see the infection, what is the infection rate, then this is the one of the best design. And they found that even after 7 months, those who had positive IgG level at baseline, only 2 developed RT-PCR positive even after 7 months. So even protection of infection, although this paper has not calculated the efficacy of protection from the infection, but the way we calculate the efficacy of vaccine, with same way if you calculate this, the protection from the infection, even after the 7 months, was almost 95%. None of these vaccines are providing even 20-30% prevention from the infection. There are other studies also. So a lot of studies. One study published in Nature. It's also a very high impact journal, and that we may SARS-CoV-2 infection, natural infection may provide lifelong immunity. That does not mean that you expose everybody, because there are some deaths. But those who recovered from COVID, definitely, whatever evidence we have, we can say they are the best protected person on this earth. Then UK also released the data of it's already available on their website, of 40 lakh people. So out of 4 million, only 53 developed confirmed reinfection, and none developed any severity. So these are the some evidences that natural immunity is providing better and longer duration of protection. The reinfections are happening, but rates are very rare. And the many reinfections are occurred during Omicron phase, and mainly because the majority of mutations are in the receptor binding domain of the virus in the spike protein. So that's why even reinfections are not only breakthrough infection. If reinfections are occurring, then definitely breakthrough infection will occur. So these are the various Israeli study. They compared vaccinated, fully vaccinated without natural infection with Pfizer Moderna 2 doses, with your unvaccinated people but recovered COVID. So COVID recovered unvaccinated, and what they found that those who recovered from the COVID, 1300% in the relative term, 13 times. So 1300% better protected if you compare with the vaccinated group from the infection. So chances of infection in these population, those who recover from reinfection, chances are very low. Although
15 minutes over. 15 minutes over.
15 minutes over. And we have given, okay, 5 more minutes. No? Yeah. So I'm going to finish in the next 5 minutes. So JAMA has published one, CDC published this MMWR mortality, your weekly report, morbidity mortality weekly report. The top one is your unvaccinated never had COVID. The second dotted line is never had COVID but vaccinated. And the last 2 line, one is, so the 4 group, never vaccinated and had COVID, and unvaccinated but had COVID, and they are protected. Although it took almost 2 years to understand that natural infection is providing better immunity. So these are the existing evidence. I'm not going to cover this. So what is the impact? See the trajectory. This is the infection rate. If they started like US, they started vaccination December 14, 2020, more than 50% fully vaccinated on August 1st. But they had several waves, and the number of deaths are also many in the US. Singapore, almost 100% fully vaccinated, but see, more than 70% August, Now it's 100%, more than 50% booster, but they had very severe COVID wave, almost break all previous record, and deaths also. Same South Korea, if you see Denmark, you see Denmark removed all the restriction here. Then everybody criticized, and they almost removed all the restrictions. And now this is the current scenario, like yesterday reported around 1,500 cases, 12 deaths. UK trajectory, just see, UK more than 50% deaths. Cases, booster they have already given here. But still, there are few deaths, and if you compare with previous year, definitely overall mortality rate is very low, and overall cases are now coming down. But they have had very severe Omicron wave. In South Korea, even today, less than 30% population are fully vaccinated. But if you see the trajectory of this, so why this is happening? We, as an epidemiologist, we must always ask why this is happening, why this is going down, down both the mortality. And the answer is they have reported that even during January time, more than 70% population had a natural immunity. Whereas now in UK, only a few days ago, around 70% of population, now they have natural immunity around 70%. So that's why all these declines everywhere is because of natural. Even before this vaccination, when we started, that time there is a threshold. I'm not going to discuss all this. See the India trajectory. After the second wave, the first wave, almost 1 lakh cases, September 2020, then April, May, 4 lakh cases per day. We started on 16th January. Then there was a gradual decline. So why this was the gradual decline? Vaccine coverage was very poor. Then also we saw a gradual decline everywhere in Delhi or in all parts of India. Deaths are also in similar way declined. This is the word. So now the current policy. Now we can say vaccines are preventing severity and death, but naturally infected are better protected than the vaccinated. So do you think those who recovered from the COVID, they require vaccination? Unvaccinated people at a risk of the society? This is the completely disinformation, I can say, even not the misinformation. Knowingly, if you are propagating a wrong information, say disinformation, both are at the now same risk of getting the infection. Unvaccinated. So it's not providing the community level protection, it's providing the individual level protection. A vaccinated person is better, safe in terms of safety from the severity and death. So better protected in terms of severity and death, but not in terms of, so vaccination against COVID will be prevent future wave, completely misinformation. 100% this vaccinated also, they had and they are having this, like currently South Korea is having. So it's a completely misinformation. They cannot prevent any future wave. They can only prevent the impact of wave if they never had natural infections. Like South Korea never had natural infection. So currently the impact is low, but still impact is there. But India during third wave, there was no impact. It's mainly because of the natural infection. Yeah. So it's finished. Now I have 1 minute. So this is my second last slide. So vaccinating children, all this. So the main message is that you must evaluate all the public health interventions with the clear-cut objective: when to vaccinate, when to not vaccinate. Unnecessarily, in my opinion, it's unethical. If you are not providing any benefit, those who recovered from the COVID, but the vaccine-associated risks are there, same. Just like booster dose, we are recommending booster dose. Already 90% people had infection. So there are some problems, even those who recovered from the COVID, all these problems, the serious adverse event are mainly due to your hyperimmune response. So level of hyperimmune response will be there. There are chances of AD also. So there are many issues. So it's not only just vaccinate. Natural infection is also providing a booster dose. So I already had provided. So evaluation of the science continue, but judicial use is important. So you must use, otherwise people will lose trust. So the policy for the vaccine must be designed after risk and benefit analysis. And if the risk is more, then we must stop. If benefit is more, we should continue that intervention. Thank you very much.
Thank you, Dr. Sanjay. You stood to your reputation of being candid and straightforward and what you think is right and giving solid data. And I have been listening to you from the beginning. So we'll bring you into the discussion a little later as the organizers have planned. And now I have my pleasure of inviting my another guru, one guru of vaccination, one guru who's a
Dr.
far excellent clinician. In olden days, Morgagni and Tony physicians, and including William Oster, they used to conduct pathologies themselves to understand the medicine. So I admire Dr. Dhruv Chaudhary because he understands the pathology, pharmacology, microbiology, biotechnology, and he's the one who treats the patient. I'm not saying just to please him, but actually I think that he's a thinker, philosopher, and clinician par excellence who can actually tell us whether the COVID vaccination has changed the game or will change the game, and how it has changed the pandemic and how it will change our lives. All to you, Dhruv. Please unmute.
Thank you, sir. Thank you, sir. You always have been very kind and gentle. And thank you. I take every word you say with all sense of humility and regards. I was listening to Sanjay. It was a pleasure. In fact, when Dr. Saurabh Kole asked me to look at it post-COVID vaccine scenario, I said, what are you looking at it? Because if I put a hyphen, it's entirely different meaning. If I take it up, it is a different meaning. But I'm very happy that what Dr. Sanjay was presenting, I share a lot of thoughts with him because we are all looking at it that probably we are dealing with a linear model, whereas the biology is non-linear. That's what one has been saying. Yes, dominantly we can expect those actions are going to be there and will make a difference. And definitely a significant advances in science has been there. So what Dr. Sanjay has shown you a solid data. What I will tell you is as a simpleton, I have just tried to put the important points which are there. And obviously they all have been taken from the data one. Secondly, before I say there is a disclaimer, is that unfortunately the solid data in terms of vaccination, except the numbers which are there, whether it is from the government of India or in the institution, long-term studies we still not been able to see except one from CMC Vellore, which they did it on some 11,000 patients. But that again was they were looking at it. But we don't have as such post-COVID one. But I'm certain as far as the post-COVID syndrome is concerned, I shall be, we have already completed one of the studies and we will be putting it in the public domain shortly. So sir, let me just first of all put my presentation. Yes. Let me share it, the screen. I have taken a bit of a liberty, so I put post-COVID, I put a hyphen and vaccine scenario, which was not there in the original one. And my thoughts are to give vaccine, not to give vaccine, all these questions which are coming to it, especially if the person has already got a COVID and how to go ahead with what does the data say. And I said in the beginning, the disclaimer is that the studies from the India are very limited. What we are seeing is the data primarily coming to us from USA, from UK and South Africa, especially from the Western world. So the only thing, good point is that at least one which has been given to large number of population in our country, that is ChAdOx, that is AstraZeneca, which we look at it known as a Covishield here. So that gives us some amount of thing, but we had a very limited data about Covaxin as well as about the other whole sequencing, the whole genomic data. Now, what we know, what Dr. Sanjay has also told, my predecessors also talked about it, is that immunity provided by a prior infection and vaccine are both high, but not complete. That means it's not 100%. So the data is enough, data is there, which he has also shown it. What is equally important is that antibody levels, they correlate with protection, no doubt about it. But what is the protective titer? Still, we don't know. How much is the level which says, yes, tells you, I have seen people having thousands of international units having it, but still getting the COVID. And the doctors in particular trying to go and get after immunoglobulin therapies. So what is the level at which we say, yes, this is one which is the threshold we still don't know, as we know for other viral disorders. Post-infection, we are looking at it varied levels of titers. Minimal, we are seeing it with asymptomatic, and more with clinical symptomatic and severe disease. In other words, as the disease severity increases, the antibody levels which we have been observing and data which is being collected is proportional to the severity. On the other hand, people who have asymptomatic or minimal symptomatic had minimal elevation in their antibody levels, but a lot of data has again coming up that quite a significant number of them also has got cellular T-cell responses, which may be protective besides antibodies. But what is very interesting is when you look at the full vaccination, it provides a more consistent level because we know at what level, how much antigen is being injected inside. Whereas when person is having an infection, we do not know the dose, though there could always be a question mark in terms of it, what is the total dose, what is the virulence, what is the immune responses, they all will be there. But when you give a vaccination, you know you have given a consistent amount of dose, and this dose, like in messenger RNA, has been found to be responsible for eliciting responses and killing the elderly people and also causing myocarditis. And that's why they have now reduced the doses from 100 mics and 50 mics, depending upon it, Pfizer and Moderna, now to 30 and 50 itself. And so that means you know the antigen load which has been given. Similarly, then elderly and immunocompromised, we all know they show poor response, whether it is infection or vaccination. Therefore, it becomes important for us to understand, to give, if we have to, let's say, give a booster dose, or we need to give reinforcement, or the so-called more diplomatic Indian way of talking in terms of the protective dose to whom. And the level of protection may not be same for all variants. So what we started from the wild variant, we came to alpha, we came to beta, we came to delta. We are now come down to Omicron, and within the Omicron, we have now going on to different variations, which is looking like an unending saga, which is continuing without realizing that the nature and the biology has its own set rules, which probably we are trying to come to terms with. Now let's look at it. I always feel as that whenever we have to do, we need to understand what the infection is calling, how we are protecting ourselves. Besides the innate immunity, it is the adaptive immune response, which is extremely important. Innate immunity is important, but I'm straight away jumping to adaptive immune responses because that is how the infections, especially the viral infections, are taken care of, and more so when you see the intracellular infections. And we know today that SARS-CoV once gets into the cell, it gets replicated, it gets transmitted from one cell to another cell. Obviously, AC2 receptors are an important function of it. What we realize is that an infection produces robust, both humoral and cellular immune responses, and whether it is mucosal or sera. We are seeing that a very high incidence of mucosal and IgA levels are there, and same is true for the sera as far as IgM and IgG levels are concerned, especially in those patients who have been symptomatic. Similarly, what we have seen, those patients who have been symptomatic, seroconversion has been up to 90%. And you can have a heterogeneity of that means when I say heterogeneity of antibodies, it can actually up to 200-fold. Some may have X and other may have a 200X. Therefore, there is a huge variability which is taking place in terms of it, and depending upon the test which you are using, whether using a camelosense, whether you are using a third generation ELISA, or whether you are using other additional methods, you can have a differential in variation. And when I'm talking in terms of these responses, I'm talking in terms of three particular antibodies. One is against nucleocapsid, another is against your RBD, what is called as a binding domain, receptor binding domain, and third, which we look at, is an anti-spike protein. Fourth is a neutralizing or the pseudo-neutralizing antibodies. We routinely don't check, but in the lab or in the research things, they have been checked. And what we are seeing is that both IgA, IgM, IgG, all they go up, and they also have a certain time period to which they but the responses depend entirely upon in the symptomatic, how severe the disease and how the immune response is. And that also tells you the variation in immune responses to individuals. And we all know that binding neutralizing antibodies are high and rise rapidly. This is what the receptor binding, as well as along with it anti-spike proteins, which we take it up, and along with the neutralizing antibody. Lower the cycle threshold, better the immune response. It simply means higher the viral load, better is the immune response. So lower the cycle threshold, that means it has been picked up very quickly in 5, 10, 15. And all of us, if you see and remember that in Delta, whereas in Alpha, when we had the initial wave, when we had the original strain and then we had the variation 614, then at that point of time in the first peak, we had the cycle thresholds coming up late 20s, early 30s. But in Delta, we were seeing a very high load and the cycle thresholds were in 16, 18, 14, 12. That's what average was on an average 18 to 20. That means it was a very heavy viral load. Asymptomatically, minimal symptomatic, I already said, have a poor immune response, and it may not be even enough to reach the threshold to stimulate immune responses. Elderly and hematology malignancy is expected. Same has been true for the people who have been steroids or who have been on chemotherapy. What is also very interesting is that neutralizing antibodies and IgG spike titers, they correlate very well with decreased risk of reinfection severity. And when we are talking in terms of anybody getting a dose post-disease, is primarily to reduce the risk of reinfection and severity. So they are the two key things. And that's what we are going to discuss in now a couple of more minutes. Whether to actually, by giving the vaccination, are we reducing the reinfection rates? And even if we are reducing the reinfection rates, is it something to do with the severity or not? If there is no severity minimum, it works like a URC. Anybody would have bothered? Answer is no. Nobody would have even bothered about it. It is because of the lung injury it caused and it affected the multiple organs and leading on to multiple effects and the post effects that we are looking forward to. And what is equally important is that not only the antibodies are important, we must understand as clinicians that cellular response is the key. No antibody will be there if the B cells don't make it. So how a B cell responds to it, whether we have the memory B cell, because it is always in the secondary infection, where if you see the secondary adaptive immune responses, when you have the second time the infection is there, and that's what vaccine is also going to do, is creating a memory B cells. So whenever the virus comes up, it recognizes through its computers, through its sensors, and immediately starts forming. And the time period which is there to make cells, memory, the B cells, which earlier takes around about 7 to 14 days, is reduced significantly. And within 48 to 72 hours, you can see the antibodies jumping. Within 1 week, they may double, treble, or even quadruple. CD8 T cells are important in killing the bacteria, and it has been shown that where you have a relatively lesser levels of quantitative values of antibody, it's the CD8 cell which has been the savior. And they have seen in rhesus macaque monkeys experiment that if you deplete the CD8 cell, and even if you give memory B cell produce enough antibodies, still they get the disease. Therefore, CD8 cell plays an important. Similarly, Th1 responses are as important in terms of stimulating the T cells and also converting some of the CD8 cells into natural killer cells. And therefore, when you see the total pattern of response to infection, it is cellular, it is along with it, not only cellular, but it is a humoral. So once it is there, these adaptive immune responses become the key for us. When it is not only the immune responses which are, their kinetics is as important. How they fare over a period of time, like you get a measles once, you don't get a measles unless you have immune suppression. Therefore, similarly, we have a lot of other viral consideration. Diseases, if they are not latent, you will see they don't come back again. So what we are seeing is that peak antibody levels after starting at 7 to 10 days, the first infection, go up to 3 to 5 weeks, and then they start waning. Whereas when you start looking at the nucleocapsid antibodies, they wane faster than the anti-spike antibodies. And that is why in the sero surveys now we are trying to go after anti-spike antibodies because their deterioration is much lesser. And at 10 months, you will see a 30% drop in nucleocapsid antibodies, whereas there will be hardly 10% drop in anti-spike, and you can pick them up in large number of patients, and you can predict positively how many had infection in the past when we started up. Neutralizing antibodies are a very interesting phenomenon, what you called as a biphasic decline. Initially, they will go up, and then 2 to 3 months down, they will come down. And then after that, they reach a level and they remain static over a period of 7 to 8 months. And then they show a gradual decline. So that is very interesting because these are the ones which, along with spike proteins, antibodies are important in protection. Moderate to severe illness, as I had talked about, have a higher antibodies up to 3 months than the person who mildly symptomatic. So at all levels, they have higher antibodies than the people who had mild symptoms. And these differences, which from studies got published up to 8 to 10 months, but now the data is showing that this stays probably up to more than 16 to 18 months. B cells, they also increase in the first month because that is how the antibodies are coming up, and their concentration remains proportionate to what we are seeing higher up to 8 months. T cells also show increase along with this point of time, and they decline and remain stable with a half-life of 3 to 7 months. They're all COVID specific. That should be very important, SARS-CoV-2 specific. And you look at it, the CD8 cells, they also stay in the charged state. They're all moving around and they continue to stay elevated up to it. That means body has been fully activated, fully charged, fully responsive to an infection if it happens again, once they have got an infection. So that means robust immune responses are there in all those people who had COVID and who was symptomatic. That is the key. The word is symptomatic. And then the protection of reinfection, that we started it up, and they found out that the RT-PCR positive SARS-CoV-2, again, this published from UK first of all, it declined by 80 to 93% for at least up to 7 to 9 months. This was the first study came up from NHS on more than 10,000 people. And what is very important is that the large number of reinfection, that 10 to 12% which was there, they were all asymptomatic or minimally symptomatic. I am sitting in front of you. I had twice the second infection. In fact, I just has a mild body ache and fatigue. And the first time also didn't have much, but that's how what we looked at the data of around 50 cases who had
50
reinfection.
minutes gone.
And then we look at it, the rider is wider, this has been, but they have to set a limitation of these studies because this data was coming up from alpha and from the delta. And the modeling suggests that immune responses continue to provide 50% protection up to 1 to 2 years as for the other coronaviruses. Therefore, it is not bringing in the question what Dr. Sanjay Rai was asking about vaccine or not to vaccine. When you look at the immune response and kinetics to vaccination, it is as good as that a natural infection. And more data has been there with the messenger RNA. I and he had the privilege to work with the Covaxin trial. So we are privy to that data, where we also saw that it stayed up to 6 months. We had a very good antibodies, and then it started decline. And they did a very smart thing because of which cellular immune responses also got activated. Otherwise, in killed, you only see humoral responses dominantly. And with obviously with Covaxin, we have been seeing additional nucleocapsid, which have been measured. Again, neutralizing antibodies have been measured, so have been the pseudo-neutralizing techniques have been done and found to be a useful test in it. And again, the data which is there, which is now in public domain, phase 3, is that it continues to stay effective up to 6 months. Quick levels here are formed because we are giving a definitive known amount of antigen quantitative amount. So quick levels are formed and you can have detectable levels of IgM, G, and A. It also induces like natural infection, long-lasting B and T cell responses. Antibodies continues to stay detected easily up to 8 months when the data has been coming up. And rate of decay matches that of convalescent patient. That is what the patients with the disease. And cellular response stabilizes up to stable 6 to 8. So if you see moderate to severe infection versus the vaccination, dominantly this data is from messenger RNA or vector-based J&J or ChAdOx, which is the Covishield. They say they probably run parallel to each other. The impact of variants obviously has shown that there is a decreased efficacy, but still significant enough to prevent severe disease is there. Mild disease again had a higher chances of variant infection along with severity, and with vaccination we see 2 to 4-fold decline in neutralization. So when you look at infection versus vaccine-induced immune responses, as I said, it is equivalent. But what is very interesting in one of the studies which came up from the USA, it showed, which was got published in MMWR, that 5.5-fold increase incidence among previously infected compared to fully vaccinated. Now, again, this is the difference in the variants which was there, and probably these and the mRNA vaccines were not good enough to protect them. Again, what it is showing is that later UK data also shows that 2 times more chances of reinfection are there if you do not vaccinate people who had the disease in the past. But as far as the severity is concerned, there is no difference. That is what is important. So it has also brought up to the hybrid immunity with the COVID-19 vaccine. If you see, we have a natural immunity. Here you will have a memory cells, you will have an antibody, you have a CD4 cells, antibodies, and CD8 T cells. Vaccine immunity also there because we are giving a definitive amount. So some amount of bias is there in terms of vaccine immunity. And this is dominantly, again, we are seeing with messenger RNA. But when you give a hybrid immunity, 2 together, they really scale up further and leading on to acceleration in immune responses. So what has happened is that if you give a single shot, this is primarily with mRNA after illness, it is equivalent to 2 shots. That means person is fully vaccinated. It also has a stronger immune response, as been shown by the previous cartoon. And this is a multiple data which is coming up that if you have an infection plus vaccination, you are having much higher antibodies level. This again is a data which has been published. If you see it, it is a ChAdX. Protection against reinfection, previously infected, if you have given is much higher. And that is how they are saying and telling that everybody who has got a disease at least should get a vaccine, though there are certain riders out there. This is again a recently published study in Lancet in March, which again is showing this is at the time since baseline, this 8 line is showing and the without immunity who had the reinfection. But if you see,
Dr. Chaudhary,
I'm just
crossed the limit. Yeah.
If you see, the decline is much faster in the patient reinfection in patients who had a hybrid immunity. And same thing has been happening with the vaccines because that though they may be messenger RNA, they may be a different vectors in the vector-based vaccine. They have a different epitopes which we have taken during the spike protein for causing and producing the vaccine. So that is why they're working together and they're working as they're enhancing each other's activity. Booster dose, because the studies dominantly have come from Israel, and later on now they have been published in New England Journal and Lancet, both from UK where they looked at different data and they found out that if you use vaccine, whether you use the adenovector, whether you use messenger RNA, or whether you use the Cobavax, which is going to be available, the subprotein, every vaccine, when we did a mix and match, had a better response. Some data has come up from India also, where people use Covishield, and when they use Covaxin followed by Covishield, they had a better immune responses. Otherwise, and similarly, they were not able to recruit as much. But the limited information which I have from the CMC Vellore, where they tried to do this study, they are less than 500 enrollment. In fact, 300 something enrollment, if I correctly remember. They found that probably using booster Covishield was far effective than Covaxin, but till it comes in public domain, I can't say about it. This is how we are seeing the booster in it. Initially, you have the gradual decline is there, and you are trying to again stimulate the B cells so that the levels go up. And if there is a certain amount of extra antibody, extra push required, you can give. That is the principles of booster, and hence we are calling it up. It's nothing else but stimulating, as we have been practicing in immunology in various diseases. But the practical problem of the severe disease immunology not properly understood. Now, can we have, if we give vaccines, antibody-dependent issues? Because a lot of these people, I also had adult multisystem inflammatory syndromes. Children also had it. Children also had myocarditis. Which vaccine to be there? They say if you have a myocarditis, shift over to vector-borne or to the killed viruses. How is it possible? Because this is the biggest experimental trial going up, and economic issues to me now, besides science, are guiding the practical current guidelines in terms of, even if I got a reinfection, if it is not severe, do I need it? An exercise in post-COVID? I think this is something which we look at it. And this is the data which was published yesterday in terms of variants, if you see effective of COVID-19 against Omicron variant. Again, you will see here is they have been trying to use combination, and the best result are actually coming with, in these cases, they are again showing with the boosting coming up with the messenger RNA vaccines after that. So the data shows it that you keep on boosting it. My question is very simple. When you are having a subclinical infections to these and already you are charged, do we really need these? And my question, it's an open-ended question, is do we really need the additional doses of vaccination? It's something which we need to look and ponder upon. So to conclude, data is in favor of vaccination after COVID, no doubt about it. Duration is still not clear when to give, how to give it. We need to have an Indian data. Majority is from messenger RNA. But whatever we are getting for the ChAdX data, it is encouraging, and risk-benefit ratio should be the key driver. And a lot of people, we have stopped not giving vaccination who had a significant multi-system inflammatory syndrome. And I don't think, and the question which I put it up when we are having a subclinical infection, which is going around, is it worthwhile or think in terms to take a vaccination which is nothing else but creating inflammation with the antigens of the virus, so that again the booster or the B cells and the T cell, they continue to remain charged and protect us. Thank you very much.
Thank you, Dhruv. I think we have had amazing four deliberations. There are more answers than questions, or more questions than answers. I cannot say that, but surely enough, the COVID vaccination has saved the mankind. Dr. Sanjay Rai may not agree with that, but if you see the global scenario, number one. Number two, there is hardly any comparison between various variety of vaccines as far as their efficacy is concerned. Number three, the natural infection gives the best immunity, and the current data say at least for 20 months. One of the papers which was published in JAMA said that it lasts for 20 months, and this information is from mRNA vaccine. Those who are infected and get vaccination get the best immunity. There's no questions about it. The fifth thing is that there is evidence that vaccine does reduce the chances of infection also. There's a whole lot of data, but however, it's uncontroversial that it definitely reduces the severity. The unanswered questions are when to give booster, what to do when the person is infected, has got a disease, when to give him further vaccination, whether to give, whether mixing vaccine is a good strategy. To take the discussion forward, I hand over the floor to Dr. Nandi. He may have some questions and comments. Thank you very much. Dr. Nandi, please.
Thank you. Thank you very much. Now, I have been listening, and I agree with Dr. Nilani, accepting that I wanted to raise some issues which we have been observing in our day-to-day clinical practice, that whether a person has got past infection with COVID and then vaccinated, or simple COVID vaccinated without prior suffering from COVID. In the next part, post-COVID vaccine scenario or situation, these individuals, when they are getting infected by some other agents, maybe viral or bacterial, whatever the infection is, it's becoming very complicated. For example, I would cite few examples. A patient suffered from COVID, got infected with say, dengue, got infected with chikungunya. And in those patients, we observed that there is a loss of balance between the CD4 and CD8 cell composition. There was a gross suppression of the CD4. As a result, what happened? The fever, which takes only 5 to 7 days to last in case of chikungunya, it went on to persist for 1 month. So this is one example only, but we have other extremes, that there was severe immunosuppression, total pancytopenia, like HLH, leading to multi-organ failures. Now, these are the issues I need that some of the people who are attending this conference may try to study this in depth and find out. This is one area I should say that this area should get its due importance. And this is one of the area I agree with Dr. Rai fully, that natural infection-induced immunity should be considered as much better than the present vaccination-induced immunity. This is our, social, community level and the clinical experiences. Thank you very much. I had posted one question in the chat box. If someone, that's almost in the same line as I discussed. Thanks a lot to all the speakers. It was a nice session. I enjoyed thoroughly, and thank you again. Thank you.
Thank you, sir. Dr. Khilani, I never said that vaccine, these are not protecting. They are reducing deaths and severity. But if you compare with natural infection is much better. But even you cannot compare, you can only compare apple with apple. Even we cannot compare natural infection versus your vaccine immunity. Really beyond any comparison in terms of severity and death, natural immunity 6 to 9 month maximum immunity they are providing, whereas natural immunity they are providing lifelong. See, the chances of getting severity is very low. And what will happen? See, all these are problems are related. Some problems are related to your hyperimmune response. So if already infected, 2 doses has taken, again infected, and you are giving booster. So what will happen? We don't know.
Exactly.
And to the best of my understanding, they will cause more harm than the benefit. This is
I agree with you. This is where I was trying to put my finger in. You see, exactly. This is my apprehension. This is my observation and hypothesis. This has to be proved.
Dr. Nandita, sorry for interrupting. Yes, I agree with you. I give you an example, my personal example. My daughter, I'm sharing it on this thing. She had 2 doses of Pfizer in Singapore. She returned back. They gave her, because as that was a part of it, she got a booster in terms of Moderna. 10 days down the line, she got Omicron, and she had all the evidence suggesting of a skin involvement. They saw skin involvement. To me, that clear that is a hyperimmune, and she has got probably secondary vasculitis, the type 3 reactions immune there. So he has to be put on steroids to actually suppress it. This is not one. We are seeing that is what a multi-system disease, which I was talking about, is that multi-system inflammatory syndrome. That is what we are seeing. And I have seen all sorts of complications which you can see with the vaccination. But the problem which we are looking at the post-COVID is where patients are having severe disease. We need also to look at it and how many cases we are also causing antibody-dependent enhancement into it. Because what we are seeing is in those who had severe, we are not giving at all. We are just trying to tell them, you hold on, we'll see, we will. And few of them infection and nothing has happened.
Dr. Dhruv, at individual level, you may be doing this, but at community level, everybody is getting booster.
Vaccination, Dr. Sanjay, when we started vaccination, there was a zero immunity. When we did a seropositivity and other surveys in Haryana in September, I give it my state, we found nearly average 76, and even in children, we were finding 75 to 72%. Between 6 to 9, 70% already had the infection. Now, when we are going with it, because we don't have a data, this is the biggest clinical trial going on. We still don't know for how long we will keep on going on. Somebody has to say, look, stop, see, and then move in a scientific way to find out do you need a booster or not. Already if you have got a COVID, you have got a vaccination. That means you already have completed the full circle. But what after that? Now suddenly we are all going after boosters. Israel has gone on to 4th. Now again, immune senescence is going to be there. That's the point I was trying to make.
Dr. Chaudhary, I feel that there has to be made some of the priority areas, at least in the post-vaccination and post-COVID scenario. We must set up certain priority areas for studies.
Absolutely, sir.
And our clinical experiences would start the ball rolling in pointing out and setting out our objectives.
Very true.
Okay. So that is the area, perhaps this is what I was trying to emphasize on you all, everyone, that we must sit together. We must exchange our views and observations. And we must come together to build kind of a area where we can undertake studies.
So, if I could come, Dr. Amitabh Nandi sir and all others, we are running late for the.
Make a comment there, Dr. Partha. Can you make a comment, please, Dr. Chilani?
Okay, sir. Okay.
No, I have no specific comment. You know, it has been amazing discussion. I totally agree with each of the participants. But when you make a public health policy, the antibody level which we are measuring is not yet standardized. And at a research level, yes, I totally agree with each point. But I think I strongly feel that, you know, a vaccination program has done a great deal of service to the mankind, and there have been some excesses. I'm sure over vaccination you can say for the purpose, but whatever the evidence is there, and I think if we are protected during the second wave, the medical fraternity was protected because they were already vaccinated. The mortality was exceedingly low as compared to those who were non-vaccinated. It is proven beyond doubt. And in the third wave, you can say that Omicron was very mild disease, but one of the things which protected us was actually vaccination / previous infection. That's my personal take, and I thank Dr. Saurabh Kole, who called me at the last minute to be part of this program. And I don't know about others, but I have immensely learned from all 4 lectures and the people that I admire all the time. Thank you very much.
Thank you, sir. Thank you all the speakers for doing very important job, and they have all done. They have expressed their topic in a very lucid and informative way. I'm also thankful to both the chairpersons, Dr. Amitav Nandi sir and Dr. Kilani sir, for offering us such a very beautiful interactive session, and we have learned a lot. So as we have to pass on to our second session of our second day World Congress on Adult Immunizations. So now from onwards, we will divide our scientific sessions in 2 halls. One is Louis Pasteur Hall, and another is Edward Jenner Hall. So there are various scientific topics are yet to be discussed. So according to I will definitely request our learned gathering to, according to their topic of choice, you please opt for either Louis Pasteur Hall or the Edward Jenner Hall. So I hereby conclude the first common session of the 24th April, second day of our adult immunization program. Now I hand over the mic to our conductor, that is Dr. Devdatta or Dr. Shatabdi Roy. Okay, over to Dr. Devdatta.
ও তো ওই যে মানে সিঙ্গাপুরের তো, আর ও তো কোম্পানির লোক, ও তো অ্যাস্ট্রাজেনিকার লোক।
Mute yourself. Someone is talking. Please mute yourself. Then have your chat. Okay.
Good evening, one and all. Welcome everyone in our session in the Louis Pasteur Hall, and now the session will be on hepatitis. I am Dr. Shatabdi Mitra, going to moderate this session. For chairing this session, we are privileged to welcome Dr. Manoj Agrawal sir. Dr. Manoj Agrawal sir is DM Fellow, DNB MRCP. Sir is Chief Gastroenterologist and Hepatologist in Bellevue Clinic, Kolkata. Welcome, sir, to chair the session. Sir.
Yeah, hello. Okay, thank you, ma'am. We have a session now on a fairly, obviously COVID has, taken everything, has become the most important thing now. But there are GI issues, and I think the sessions today are on typhoid, which continues to trouble us all along, and also on hepatitis. And hepatitis, we have all seen, as we have from a time of medical college till now, how the development has happened in terms of vaccination, in terms of protection for the rest of the people, and it was posing a huge problems, particularly hepatitis B. I mean, it was one of the leading causes of morbidity and even mortality, and some of the malignancies. So I think we have got very good speakers with a lot of experience in their own fields. And I think we should, without wasting any more time, let's go ahead. I would like to invite Dr. Reena Ghosh, who's the first speaker. She's a pediatrician with the Institute of Child Health, Kolkata, and she's in charge of the immunization program there. And she's trained in advanced virology and has been a co-investigator in several vaccine trials. and has several publications to her credit. Dr. Reena Ghosh.
Thank you, respected chairperson. Good evening, everybody. I would like to know whether my slides are showing or not from the technical person.
No, ma'am. Your slides are not visible.
1 minute.
And now it's visible, ma'am. Just make it full screen.
Okay, thank you. Okay, thank you so much. Am I audible?
Yes, ma'am.
Okay. So, for typhoid fever vaccine. Under this typhoid fever, we know it is a very important public health problem globally, causing significant health challenges especially in low and middle income countries. So in India, also there is a substantial burden, and also there is some concentration.
Ma'am, your slides are not visible. Can you share and unshare again?
Okay.
Now it's visible.
So I will just, this is an important public health problem globally as well as in India, as India bears the half of the global cases. And we all know that it is an acute febrile illness caused by Salmonella enterica salivary typhi, which is commonly known as Salmonella typhi. What is the epidemiological importance of the typhoid fever? The most important is that an attack of typhoid fever does not provide any long-lasting immunity. So the person who has already suffered from typhoid fever can be reinfected again and again. The most important is that who are the convalescent carrier, they will keep on shedding the bacillus in their urine and stool for months together, even up to 1 year, because they harbor the bacilli in their gallbladder also. So carrier state remains in 3 to 5% of the already who are suffered from enteric fever. And the importance of this carrier is that this carrier may infect the other family members of the same family, and also those carrier may remain asymptomatic. So they will remain asymptomatic for years together. We all know the typhoid Mary incidence. Or this carrier may infect the other persons, so they may get actually the enteric fever. Another important aspect of this typhoid fever is that, so there is a recent outbreaks which is showing the alarming trend of drug-resistant and multi-drug resistant of typhoid strains. And the development of the H58 haploid of the typhoid strain in Pakistan in 2015 is extensively drug resistant. It is resistant to all the primary antibiotics which are being used for the enteric fever, and also this H58 typhoid strain is also resistant against the ceftriaxone and fluoroquinolones also. So it will be very difficult to treat these resistant strains, and we might in fact go back to the case fatality rate as far as case fatality is concerned to the pre-antibiotic era. So it is a very grim situation. And also other epidemiological factors, that is the rapid urbanization, which has outdated the inadequate or unsafe sanitation system and has increased the risk for the typhoid transmission. And the most important is the climate change. We know that the global warming, which is the higher likelihood for the natural disaster, if there is a drought, there will be shortage of drinking water, and the people will be forced to drink any water which is available and which is likely to be contaminated. And on the other hand, if there is an flood, So all the water systems which are being given for the drinking purposes may be also contaminated. So climate change is also very important, and which by all means we are not able to yet control. So infects the typhoid fever. Let us see the typhoid fever burden. It infects roughly the 21.6 million people and kills an estimated 2 lakhs of people every year. And this is a very recent data as shown in 19th August 2019. And half of these cases, that is about 14.3 million of global cases of enteric fever in 2017, has occurred in India only, where we have found that 8.3 million cases in India alone and 72,000 deaths. Therefore, also India is called Typhoidistan. So national incidence, which is absolutely a recent one, it is in the November 2021, has shown that 360 cases per 1 lakh person per years, and which has caused the 8,930 deaths and assuming a 0.2% case fatality rate. As we have seen with the other diseases also, the India is studded with all red excepting few pockets. And it is a high burden disease country. What is the high burden? Where the 100 cases occur per 1 lakh population per year, then middle burden cases are 10 to 100 cases per 1 lakh population per year, and low is less than 10 per 1 lakh population per year. So amongst the high burden countries are South Central Asia, Southeast Asia, and the most of the burden is borne by Bangladesh, Pakistan, and India, and also Nepal. And medium burden country are rest of the Asia, Africa, Latin America, Caribbean, Oceania, except New Zealand and Australia. So let us see, there are incidence rate of typhoid fever in Asia. This is a comparison of two multi-center trials. We have seen that the Pakistan is topping the list, is having 451.7 cases per 1,000 population. And there is a comparison between the 2005 and 2008. It is not at all decreasing. The incidence per 100,000 population, that is per 1 lakh population, is supposed to be decreased as we are advancing, we are being developed, we have got good sanitation control and good drinking water supply. On the contrary, in 2005, it was 394.2 in Pakistan, which has increased to 451.7 in 2008. And in India, in 2005, it was 136.7, which has increased to 214.2 in 2008. So there is actual increase in the number of cases of typhoid fever. So let us see the incidence amongst the adults. There are some studies, although we know the maximum burden of the typhoid disease is borne by the children below 16 years, but there are certain studies which have shown that above 16 years also, there are quite considerable burden of the disease also. Like in Calcutta only, there is a study which has shown that above 16 years, there was 119.7 cases per 1 lakh population. And similar study was done in Indonesia, which has got less burden of the disease, and Pakistan is not available, and Vietnam is 10.9. So let us, before going to the vaccination, let us again go to the structure of the Salmonella typhi. We all know that Salmonella typhi has three antigenic structure. One is H flagellar antigen, and there is somatic antigen O, and this is the third one is the VI capsular polysaccharide antigen. This capsule imparts the virulence as well as the antigenicity and the immunogenicity of the vaccine. So what are the vaccines we have available in our hand? The whole cell kill inactivated vaccine, because of increased reactogenicity, is no longer being used, and this is the first generation typhoid vaccine. And other three are live attenuated vaccines, VI capsular polysaccharide vaccine, and VI capsular polysaccharide conjugate vaccine. So we have got first generation vaccine, whole cell kill inactivated vaccine, which is no longer being used, and other three TY21A, VIPS, and TCV, all are second generation newer vaccines. So let us now look at the vaccines. This is the live attenuated vaccines TY21A. It consists of a mutant strain of Salmonella typhi type 2, TY2. The immune response is adequate and starts from 14 days after the inoculation. And 3 doses of vaccine, it is enteric coated capsule, is given orally on day 1, day 3, and day 5. But nowadays, there is another regime where the 4 doses capsule are being administered on day 1, day 3, day 5, and day 7. And it confers 67% of the protection over 3 years and 62% protection over 7 years of follow-up. It is very safe. There is only mild GI disturbances like diarrhea, vomiting, pain, abdomen. And next, common symptoms are general symptoms, systemic symptoms, amongst which is very common is fatigue and myalgia. And also there are some cases of fever also. So what are the contraindications of this vaccine? As this vaccine is a live attenuated one, it cannot be given to the immunocompromised persons. As it is a capsule form, it cannot be swallowed by children below 6 years, so it should not be given below 6 years. And also it is prudent to avoid this vaccine in debilitated elderly persons with chronic health conditions. And pregnancy, as because there is no trial, should not be used, these live attenuated vaccines. And moreover, as it is a bacterial vaccine, we know that antibiotic therapy along with the vaccine will diminish the effect of the vaccine, so not to be administered when a person is on any antibiotic therapy. And actually this is basically a traveler's vaccine, and mainly the persons who are traveling from the developed to the developing countries where there is high burden of the typhoid fever, they also take the anti-malarial drugs. So this TY21A should not be given along with the probe 1. And it is also recommended for travelers visiting the high burden countries, especially India, Pakistan, China, Sri Lanka, Nepal, and other high burden countries, which I have already shown. Now, this new generation vaccine is, this is the parenteral vaccine. The VI capsular polysaccharide vaccine, VIPS. It is an injectable subunit vaccine, and it gives protection between 55 to 72%. But the disadvantages of the polysaccharide vaccine is that as it is T-cell independent and does not produce any immunological memory, cannot be given in children below 2 years of age. And being a polysaccharide vaccine, having no immunological memory, has to be repeated every 3 years. Then what is the next choice? Now we have got a very good vaccine, that is the VI capsular polysaccharide conjugate vaccine, that is the TCV. This is first licensed in India in 2013 for parenteral use, that is for IM injection. The dose is single dose, not to be repeated anymore, 0.5 ml of IM injection. It can be given in children above 6 months of age and all adults up to 45 years of age. As we have seen, that adults, they contribute considerable typhoid burden in the society. So adults should also be vaccinated. 4 BIPS conjugate vaccines are licensed in India and 2 are already WHO pre-qualified. There is improved GMTs and seroconversion rates compared to the VIPS polysaccharide vaccine only, and antibody develops 2 to 3 weeks after the inoculation. So this is a chart showing the new generation typhoid vaccine. So what are the new generation typhoid vaccine? We have got oral. Of course, this typhoid is not available in our country, and then the parenteral vaccine. So we have VI polysaccharide vaccine. Then we have got the VI polysaccharide conjugate vaccines. As I already mentioned, that we have got 4 VI polysaccharide conjugate vaccines. The first one is conjugated with Pseudomonas aeruginosa exotoxin A. It was developed in Vietnam, VI-REPA. This is called VI-REPA. This is not available in India. This is the first conjugated vaccine which was tried in Vietnam. The next came the vaccine in India that is conjugated with tetanus toxoid, 25 microgram. And this is the conjugated with CRM 197. And another one is also conjugated with tetanus toxoid, only 5 microgram. So recommendations of typhoid fever vaccines. It should be used for the control of endemic typhoid diseases, and it is always used as outbreak control measure, and it gives quite satisfactory result. And high risk populations to be vaccinated, as I already mentioned, travelers, and they should be vaccinated at least 1 month before their travel. But if they are planning, if they missed 1 month, but even within 15 days, it will give quite a good protection level. And also microbiology laboratory technologists, they also to be vaccinated because they are at a high risk. And the last but not the least, the family members of the typhoid fever sufferer who have already recovered from the typhoid disease, their family members should be vaccinated, as we have seen that these typhoid bacteria will shed continuously in stool and urine for a quite a long time and will infect easily the family members. So what should be our strategy to control typhoid fever? The development of infrastructure, of improvements in water and sanitation, that is the hygiene, wash. These are the key components to an integrated strategy to control typhoid fever. But this development of this infrastructure to build up will take decades in low and middle income countries. Now we are having a safe and effective vaccine should be introduced in low and middle income countries to minimize the burden of the typhoid disease, which is not at all negligible, which I have already mentioned. Thank you.
Thank you, Dr. Ghosh. That was very crisp and clear. And, we obviously typhoid is a big burden, and I think we should give a thought to vaccinating people. Do you need to vaccinate somebody who's already had typhoid?
Definitely. Definitely, who has already had typhoid will wait 6 weeks, and after that we'll vaccinate.
Okay.
That is a routine practice. That is a routine practice. If a laboratory confirmed, may it be laboratory confirmed enteric fever or may it be clinical enteric fever, should be vaccinated after 6 weeks.
Thank you. I think if there are any questions, you can put it in the chat box and then maybe we can take it at the end. I now request Dr. Indrajit Tiwari who will be talking to us about his experience about hepatitis vaccination, hepatitis B vaccination in patients going for liver transplant. Dr. Tiwari is the head of gastro-critical care at Apollo Group of Hospitals, Kolkata. And he is an anesthesiologist, but has been closely working with GI and liver patients. And I would request Dr. Tiwari to please share your experience about hepatitis B vaccination in this group of patients. He's had several publications related to different fields of gastroenterology and some of the interventions which they have actually done safely in many of the patients. And now Apollo, they are doing the transplant in East India on a fairly regular basis. So let's listen to Dr. Tiwari. Thanks.
Thank you. Thank you, sir. Thank you, Dr. Manoj Agarwal, sir. Am I audible there? Am I audible there? Anybody?
Yes, yes.
Yes, yes.
Thank you. And my slides are visible there, no?
Yeah, everything is visible.
Thank you. Thank you. My topic is very simple. I have chosen this topic because, as Dr. Manoj Agarwal sir has said, we are now regularly doing the liver transplantation, and we come across this topic very commonly. When you get any patient who is evaluated for liver transplant in the waiting list, and when we got that this person is not vaccinated with hepatitis B, it's a big surprise to us. So, and it's such a clinical implication as well. So I have chosen this topic. Basically, this is a paper from Tony et al. And I have put this topic to aware our hepatologists, our physicians, how important it is and why should we vaccinate all those patients who are in a waiting list or patient with advanced liver disease with or without cirrhosis. So basically, although the liver transplantation is considered to be a high-risk procedure, as we know, it is a well-established as a treatment option for the cure and quality of life enhancement for the individuals who suffer from the diseases, that is end-stage liver disease, acute liver failure, and hepatocellular carcinoma. Many of them are linked to hepatitis B virus, we all know. On the other hand, transplantation may bring about a risk of disease transmission by hepatitis B virus in the periods before, during, or after the procedure as well. So, preventing an infection by hepatitis B virus through immunization schedules has been the most effective way to reduce the complications, since it decreases the number of people who suffer from chronic hepatitis caused by hepatitis B virus and eradicates its transmission in general. So it is very, very important. I want to highlight this. And this hepatitis B virus vaccine is highly effective in healthy individuals, whereas it is little immunogenic in immunocompromised individuals, including those who have not been transplanted, but who have advanced liver disease, such as chronic hepatitis C, along with B. Thus, the specific immunization schedules are recommended for the specific group, and it is considered to be the first vaccine with an impact against the cancer, taking into account that hepatitis B virus prevention reduces the risk of cancer linked to that virus. Besides, we know it is the first vaccine against a sexually transmitted infection as well. So I think I'm clear that how important it is in preventing the chronic liver disease due to hepatitis B, progression into hepatocellular carcinoma, and at the same time, prevention of STD as well due to hepatitis B. Now, the hepatitis B infection in this CLD patient. We know it is a well documented that co-infection with hepatitis B and hepatitis C is associated with an increased risk of cirrhosis as well as hepatocellular carcinoma. And a study that looked at the liver histology in patients with chronic hepatitis B and C found a higher prevalence of moderate or severe chronic hepatitis or cirrhosis, that is 63% they got out of 65%, compared to those with hepatitis B alone, that is 47% of 90%, or chronic hepatitis C alone, that is 41% in 98%. So the co-infection is having the higher risk. And patient with co-infection had higher level of serum aspartate transaminase, suggesting a more vigorous immune response as well. So data on the superinfection with hepatitis B in those with CLD are limited. There are isolated reports of adverse outcomes, including the fulminant hepatitis, following acute hepatitis B virus infection in patient with chronic hepatitis C infection. There is a small study, that is a case control study from Italy, found that patients with chronic hepatitis C infected with acute hepatitis B virus are at increased risk of developing ascites, hepatic encephalopathy, compared to those with acute hepatitis B virus alone. So most of the study is favoring that if it is a co-infection, the recurrence, the decompensation is more common. Now we come to the immunogenicity of this hepatitis B virus in chronic liver disease patients, CLD patients. So hepatitis B vaccination is recommended for all the patients with chronic liver disease, with or without cirrhosis. Currently, both single antigen vaccine like Incidence B, Hepatitis Recombivac's HB, and combination vaccine like Twinrix, that is hepatitis E and B vaccines are available. Although these vaccines are highly immunogenic in healthy individuals, that is more than 90%, multiple studies have shown that factors such as advanced age, male gender, obesity, diabetes mellitus, and chronic kidney disease are associated with a reduced immune response. So we have to be careful when we are dealing with this special subgroup of patients. Similarly, the seroconversion is poor in those with CLD, and vaccine efficacy wanes off with the increasing severity of liver disease. So if you are vaccinating your patient in early part, it has a better immune response. A study that evaluated the efficacy of 3-dose vaccine in cirrhosis have reported highly variable seroconversion rate, that is anywhere between 16% to 79%. So it's a very much variable. So you cannot rely and you have to test a titer when you are considering for transplantation, at least in your patient. So although the high quality randomized prospective trials are lacking, it is safe to say that vaccine efficacy in CLD is suboptimal without any doubt. Even the multiple strategies have been proposed to improve the immunogenicities, such as accelerated vaccine regimes and increased vaccine dosing. Because in sometimes some patient waiting for the liver transplantation or someone in the waiting list, these patients at very short time. Sometimes we get a month time, 2 months time like that. In that case, we can apply this either of the regime, that is accelerated vaccine regime or increased vaccine dosing. That is, if you go through the study, one study compared the standard dose vaccine of Twinrix and Incidence, 97 serotic patients, that is 20 microgram at 0, 1, and 6 months, with a 40 microgram booster of if patient was non-immune, against a high dose accelerated vaccine. They have taken in 51%. High dose means double the dose, that is 40 microgram at 0, 1, and 2 months, with the schedule repeated as a booster in those who are non-immune. And they found that seroconversion in the high dose group was only 45%, while in the standard group, it was 51.5%. However, when a booster dose was given, there was a significant improvement in this high dose group, and vaccine efficacy went up to 79%. That means if you are considering in a short-term basis, you have to give what we do, you administer in a high dose regime as well, you can. And a recent study found that the newly approved, that is 2-dose vaccine, like Hepatitis B, offered the highest sero protection, that is 63% versus 45%, a significant p-value in chronic liver disease compared to the traditional 3-dose vaccine. And patients who received this Hepatitis B were almost 3 times as likely to achieve immunity compared to those who received Incidence B after adjustment for the comorbidities. And we have to keep in mind that patients who had cirrhosis, COPD, or renal failure were at higher risk for pure immune response. So now we have discussed the thing. So now the simple intervention, what my topic was, how to intervene so that the adherence will be better. Because we know there is an unmet need to improve this hepatitis vaccination status in the patient with chronic liver disease. Primary care physician and outpatient hepatologist often fails to vaccinate as recommended. what they have found in multiple study. That is, in routine practice, we miss the thing, or we are unable to convince the patient the importance of this hepatitis B vaccination. Thus, we find that new strategies to improve the hepatitis B vaccination rates are required. And that simple technique in that paper, what they do, they initiated this hepatitis B virus vaccination during the hospitalization routinely, and a detailed recommendation on a subsequent vaccination in the discharge letter improve the previously inadequate vaccination rate in the OPD setting. That means we have to make a rule that every patient admitted in your ward or getting into your OPD or visiting a hepatologist or gastroenterologist, you have to keep in mind, if your patient is not vaccinated, or even it is vaccinated and titer is low, it's our duty to vaccinate it properly. And similar measures should be implicated, because this was a very, very simple step, and they found that REMS was very, very good. And similar measures should be implemented at the earlier time point of the chronic liver disease to achieve the higher immune response rate. Thank you. Thank you very much for your kind hearing. Thank you, sir.
Thank you, Dr. Tiwari, for telling us about a very important aspect of vaccination. I would just go on to take this a step further. Actually, any patient with any chronic disease, not just liver disease, it could be lung disease, it could be renal disease, even diabetes, people who are coming in touch with the healthcare system again and again, every physician, whether you're a hepatologist or a gastroenterologist or your own specialty, or even a physician, a normal family physician, you must look at opportunities to vaccinate not just against hepatitis B, but many other, for example, say pneumococcal vaccine. We know that, sepsis ultimately seems to be a giant killer. A liver patient who's fairly stable for years together suddenly develops a pneumonia or some infection which is possibly vaccine preventable and lands up in a big soup with acute on chronic liver failure and may even die. So every opportunity should be taken by every physician to vaccinate these adults, because we don't think so often about vaccinating adults. But these are people, some of these diseases are preventable. And I think a checklist like what you mentioned, initiating something when they're in the hospital or when they're visiting the clinic, and even having a checklist for adult vaccination in all chronic liver disease patients would be very useful. Thank you.
Yes, sir.
I think if there are any questions, I think you can put it in the chat box. We'll again move on in the end. And then we have one more topic to be covered. And we have got Dr. Anirban Doloi, who is the Assistant Professor of Community Medicine at RG Kor Medical College. He's been particularly involved in vaccination awareness and training for hepatitis vaccinations. And he's got several research papers. And, they have been doing some coordination work with other institutes regarding vaccination and community awareness. So Dr. Anirban Doloi, he will take us through the hepatitis B vaccination, the journey over the last 30 years. Unfortunately, we still don't have a hepatitis C vaccination, which is also a big killer. But obviously, COVID vaccination, the RNA, being an RNA virus, I think it might actually provide new insights into this area. So Dr. Doloi.
Good evening, everybody. Hope my screen is visible to everybody.
Not able to see your screen.
Okay.
Can you please share screen? Yeah.
Is it now?
Yeah, now it's visible.
Okay, thank you. So, thank you so much, the organizers.
Sir, excuse me,
the
sir.
chairperson.
Sir,
Yes.
your screen is not visible. Just try to unshare and select the first option, screen. Unshare, sir. When you are sharing, at that time, the first option, the screen is there. Just select that.
Is it coming now?
Yes, sir. Just make it full screen. Now it's coming.
Thank you. Thank you. So I will not waste any more time. So thank you, the chairperson and the respected organizers. So hepatitis vaccine and journey in last 3 decades. So it's actually a vaccine with so much importance in our life, and working. I don't want to start my video because there is a network issues. Thank you. Deaths as per caused by the hepatitis B and C infection is total worldwide is very high, and the virus is almost commonly transmitted from mother to child during birth and delivery, as well as to the contact with the blood and other body fluids. You all know about that. So I'm not going into the detail of this. So the history of hepatitis B vaccine, if I consider, there is a first generation, second generation, and the third generation. Specifically, this is an history-based thing, or which we can get the knowledge about this hepatitis C vaccine, which don't come into the play and at a regular basis, but we can know, we can have this information for our knowledge purpose. So first generation is basically plasma derived against the HBs antigen and the pre-S surface antigen. And the second generation is basically yeast derived. Also, this is also recombinant. But in the third generation, which is generally using right now, is a mammalian cell derived recombinant vaccine, which is usually we are using. So in Indian context, if I consider it, that hepatitis B vaccination has been incorporated in the year of 2002 to 2003 in the pilot basis in 14 cities and 33 districts in India. Later on, the 10 more states will be has been added, and as well as the entire country has got the opportunity of universal immunization program and the hepatitis B vaccination in the phase 2, that is in the 2011-12. So it took almost 10 years to get into the whole nation. And initially it was a 3-dose schedule, and later it changes to 6, 10, 14 weeks with an additional dose being given within 24 hours of birth, which I generally call it as a birth dose. But today, as this is a session of adult vaccination, adult immunization, we will not discuss in that detail. So this is actually the GenVac, which is commonly used in our country, and this is a recombinant one, and it produces produced from the antigenic proteins of the hepatitis B virus by recombinant process that duplicates in the chemical messages and secreted factor, that is interleukin 2. And basically, if we consider the indications among the Indian adults, then we must categorize it into 2 to 3 things. Like, first of all, the person at risk for the infection by sexual exposure, person at risk for the infection by percutaneous or mucosal exposure to blood, and others. So if I consider, there is 6 spokes, we can say. The first of all, the household contacts of HIV positive patient, as well as the current and recent injection drug users, and the residents and the staff of facilities of developmentally disabled person, the person with diabetes aged 19 to 60 years or more than 60 years, hemodialysis patients and pre-dialysis, peritoneal dialysis, home dialysis, etc. So there is a high chance of mucosal contact as well as blood transfusion, which can get you the infection. But most importantly, what I want to emphasize is the healthcare and the public safety personnel with a reasonably anticipated risk. I think in the session by Dr. Kaushik, he will discuss the adult vaccination for the healthcare workers. So he will discuss in detail also. But for my purpose, the hepatitis vaccine, I will describe. So if I consider, as Dr. Tiwari has also already mentioned, that the person with chronic liver disease am I
We've lost the screen.
Yeah. Dr. Duli, we are probably offline.
no, I am here. Hello.
Okay, but your slides are not visible.
Okay.
Probably you have to unshare, then again you share. Yes, visible. Please make it full screen.
Yeah. So if I consider the dosage the pediatric dose vaccine, we all know about that. It is incorporated into the immunization schedule, universal immunization schedule. But in adult dose, it is 20 microgram dose with 1 ml suspension and is recommended for adults aged 20 years and above. And if I consider this schedule, that is for the children, it is 0, 1, 6 months. And so for the infant, it is 6, 10, and 14 weeks, along with the birth dose, and 0, 1, and 6 months for children, adolescent, and adults, as well as for the protection. This is recommended by WHO. So if I consider the journey of hepatitis B vaccination, we must get the information about immunogenicity and the efficacy of the vaccine. That is, we can get the information that 90% of the infants, children, and adolescents, and more than 90% of the healthy adults less than 40 years of age has developed the protective antibody response following a complete hepatitis B vaccination series. The hepatitis B vaccination is 80% to 100% effective in preventing an infection or the clinical hepatitis in those who received the complete vaccination series. And the most important issue is the vaccine-induced antibody levels decline with the time. And if I consider the safety and side effects, more than 1 billion doses of hepatitis B vaccination has begun worldwide, and it is considered as one of the safest and most effective vaccines ever made. So still now, there is no evidence of hepatitis B vaccination causing sudden infant death, autism, or multiple sclerosis like that. And the common side effects are actually the soreness, swelling, redness at the injection site. This is very common to any kind of vaccine. So if I consider that is first kind of anti-cancer vaccine is the hepatitis B. And adults and children with normal immune status, booster dose are generally not recommended. This is most important because every time we are getting the patients, we are getting the healthcare workers for the suggestions that if we have got the vaccination in our childhood, we have got the vaccination in our adolescent phase. So should I need any kind of booster dose? You need not. So you cannot get hepatitis B from the vaccine. That is one of the most important information for the general population. If you have a current hepatitis B infection, that is your HBs antigen is positive or have recovered from any infection, the hepatitis B vaccine series will not benefit you or clear the virus. However, the vaccine can provide you a lifetime protection for the loved ones who do not have the hepatitis B and get the vaccine as soon as possible. That is actually the recommendation for our side to the patients. The pre-vaccination screening and serological testing, if I consider it, then pre-vaccination screening in the general population has not been found to be cost-effective in India, so we are not going into the details. The post-vaccination screening is not indicated routinely except in immunocompromised patients, sex partners of the HBs antigen positive patients, healthcare workers at risk for continued percutaneous or mucosal exposure to blood or blood-body fluids. It is actually this kind of awareness to be raised, should be raised in our country, in our healthcare setup, in our institutions, so that we can get accustomed with this hepatitis B vaccination and regular follow-up. The non-responders who are actually your HBs antigen and anti-HCV negative should receive a further full course of vaccination as 4th, 5th, and 6th dose. This is an exceptional case. If there is no response, there's 40 microgram of recombinant vaccine is administered at 0, 1, and 6 months. Retesting should be done 1 to 2 months after the last dose. So if the person remains in a non-responder, alternative strategies of protection must be explored. And the most important factors affecting this response, immunogenicity or immune response, are the vaccine factors, source factors, and obesity, diabetes. There is comorbidities. You all know about that. So we're not going into the detail. So in the prevention part, if I consider, the vaccine is one of the most important, along with the infection control, safe blood strategy, safer sex, etc. So I will not go into the detail. This vaccination is that the hepatitis A vaccine. I want to give emphasis on that. Hepatitis A vaccine, this live attenuated vaccine or killed inactivated vaccines which are available right now, HAV we are saying, and this is a single antigen vaccines, and combinations also HAV and HBV are both antigens are usually we are getting these combinations. The HAV vaccination may be unnecessary in northern India. This has been a study done by this Verma et al. That because of the seroprevalence of the protective antibodies against HIV in the school children aged over 10 years of age remains above 95%. So there may be a question that anti-hepatitis A virus antibody or antigen or the immunization is whether it is required routinely or not. So this is there may be a question. So indications are, this is actually taken from the WHO site. So persons with chronic liver disease means men who are having the sex with the men, and the persons who use illegal drugs, persons infected with other hepatitis virus, who receive clotting factor concentrates, who have received or are waiting for the liver transplant, food handlers, single antigen vaccine formulation should be administered. But most likely, preferably, these are not used in India. Still, as this is a platform for the other speakers, from the other states, other countries are participating. So I am giving this kind of details. And this is one of the most important area, that is combined hepatitis A, B, and few companies had launched this kind of combined vaccines for hepatitis A and B together in India. But still, this is not very much familiar. There's not very much available in India. There is administration can be done in the 3 doses, 0, 1, and 6 months, and alternatively a 4-dose schedule can also be done with a booster dose at 12 months of age. And adult patients receiving hemodialysis or with an immunocompromised state should receive 1 dose of this recombinant vaccine. Now, if I consider these kinds of post-exposure prophylaxis, this is a most important slide, I think. So there is documented responder after complete series, that is 3 or more doses. So in cases of these people, these healthcare workers, there is no need of hepatitis B vaccination or any kind of prophylaxis or any kind of testing or any kind of your serological testing. If there is documented non-responder after 2 complete series, that the patient is known positive and known negative, you are getting this kind of information that if there is negative, no action needed at any point consideration. But if the patient is positive or unknown, the 2 doses of immunoglobulin separated by 1 month is needed, but the post-exposure vaccination is not recommended. The response unknown after a complete series, if the positive, negative, or any result, then you must go through this slide, because if this is positive, then less than, you need to get this kind of post-exposure testing result for a HCP, that's anti-HBs, so healthcare professionals. So these kind of, those who are at risk, these kind of regular follow-up, regular test of antibody in the blood level, that should be done. and it is recommended from the, if I consider our national program, it is recommended. And unvaccinated or incompletely vaccinated or vaccine refusers, you know what to do. This is mentioned over here. Complete vaccination is necessary with immunoglobulin. So this is a prophylaxis, post-exposure prophylaxis or occupational exposure prophylaxis. I think Dr. Nag will give into the detail. And this is the non-occupational exposure. If there is any kind of non-occupational patients who have written documentation of the complete hepatitis B vaccination series and do not receive post-vaccination testing should receive a single vaccine booster dose after non-occupational exposure to an H-based antigen positive source. So these are actually not for the regular healthcare workers. These are for the extra non-occupational exposure. What I say that is the emergency or any kind of barrier failure like that. The persons who are in the process of being vaccinated but who have not completed the vaccine series should complete the vaccine series, receive the appropriate dose of immunoglobulin as soon as possible. An unvaccinated person should receive both immunoglobulin and a single dose of hepatitis B vaccine as soon as possible after exposure, that is preferably within 24 hours. So this is the actually my slides, my presentation, and to brief, if I want to brief the presentation of the my lecture, I must say that if I consider these healthcare workers, they need the vaccination on time, as well as there is a post-exposure prophylaxis where the vaccination is a very important tool. And next is the high-risk population should be identified and they should be vaccinated, as Dr. Tiwari, Dr. Indrajit Tiwari, our chairperson has already mentioned. This liver transplant patient, any kind of repeated blood transfusions are going on in patients with repeated blood transfusion, drug abuser, the hepatitis positive, hepatitis B positive patients, life partner, they should also be vaccinated. And the vaccination is complete. We can say this vaccination is complete or completely vaccinated only when the person can get the patient got this whole vaccination 0, 1, and 6 months. And if there is immunocompromised, we have to give another booster depending upon the situation, depending upon the severity of the disease or severity of the threat. So thank you so much. I think there is a time constant, so I was a little bit hurry.
Thank you, Dr. Dulai. I think this is an important topic, actually, the development of vaccines. I would just like to mention that, for hepatitis A, you mentioned on paper, but the epidemiology of hepatitis A has changed to some extent over the years because of better hygiene and sanitation and water supply. So what we do find is, young adults coming in with acute hepatitis A, and some of them tend to get a more prolonged illness, a cholestatic form of hepatitis A, and it causes morbidity and, away from work for a long period of time. So maybe in some parts of the country where they still find 95% seroprevalence, but in the rest of them, if they can afford it, I think it should be hepatitis A vaccine can be offered to young people as well, because maybe they have not been exposed and not had an asymptomatic infection,
Yes,
in
sir.
the early childhood. So this is something
sir.
to consider. I
Yes,
think China
I want to add something, sir, to you to this comment, because there is a study going on in the northern states, in the rural population as well as in the urban slums. So whether there is a prevalence, we can find the prevalence, and then we can recommend the government for the vaccination and everything. That is, it is funded by the government also, the central government also.
Okay. I think regarding hepatitis E vaccine, I believe China has developed, because A and E are almost similar illnesses, and then E, particularly in pregnant women, can have a very dangerous sort of course.
China, yes, sir. China has developed a very good vaccination of series. I have not gone through the details, because there's HBE against the hepatitis E antigen against also different types of hepatitis B surface antigen, hepatitis B your capsular proteins. There are several kinds of, they have developed so much of vaccines in their country, but the thing is, in their immunization, in the adult regular immunization schedule, they have not introduced any of them till now. So this is a big fallacy. I don't know why this was done, but this needs to be clarified.
Particularly in ladies, this would be very important in the pregnant age group, because if you get hepatitis E in pregnancy, and we have epidemics in our country, we have seen cities being affected by hepatitis E. So this is something important. And Hep C, of course, I don't know why, but this virus has been around for almost maybe we know about it for more than 30 years now, and we don't have a vaccine for it. And this was one area where a vaccine would really help. Fortunately,
Yes,
the treatments available are very good now for hepatitis C, and you can even treat it just like tuberculosis or something, where the treatment works very well, and short treatment is there now for 12 weeks to 24 weeks. So that is the development which has happened there, but I think vaccination is still need of the hour for hepatitis C.
Yes, sir. Yes. Thank you.
Thank you. Thank you so much. So if there are any questions in the chat box, if the organizers can put it. Otherwise, we'll move on to the next topic.
Thank you, sir. Lots of comments has come in the chat box. That is, the session is good, but no such question is there. Only one question is there, sir. If one person has taken the hepatitis B vaccine, single dose 4 to 5 years back, is it necessary to take the vaccine now?
Yeah. Okay, Dr. Dolai, would you answer that? Yeah.
So so you need to complete the whole schedule, that is 0, 1, and 6 months. If there is a 1 year of gap between or incomplete vaccination, like in this last case, the person has got only 1 dose, the 5 to 6 years back. So he or she needs to complete the whole the vaccination course, that is 0, 1, and 6 months, and that after that he or she can be considered as a completely vaccinated. But considering the last dose, we should initiate the whole vaccination right now. We cannot consider this the previous vaccination as one of the dose or one of the component of the whole vaccination process.
Thank you, sir. Thank you, Dr. Manoj Agarwal, sir, and all the respected speakers, Dr. Rina Ghosh, madam, Dr. Indrajit Tiwari, sir, and Dr. Anirban Duru, sir. Now we are going to another session, that is special group vaccines. For this session, we are requesting Dr. Ujjwal Sengupta, sir, please to chair the session. Dr. Ujjwal Sengupta, sir, is past president of Bengal State Branch of INA. Sir has been trained as a TOD by Government of West Bengal in the Corona vaccination drive. Sir is recipient of different national and international awards. Sir is a very known person and very known figure to all of us. Sir, please chair the session. Dr. Ujjwal Sengupta, sir. Sir, please unmute yourself. Sir, please unmute yourself. Ujjwal, sir, please unmute yourself. Yes, sir. Sir, please unmute yourself. Sir, you are not audible. Yes, yes, sir. Now it's okay.
Shall I start the thing?
Yes, sir.
Yes. So today there are three topics. The first topic is immunization of adolescents. And this speaker is Dr. Mohammad Shahidul Rahman from Bangladesh. He is the pediatrician of Dhaka Medical College. And the second topic is immunization for healthcare workers, HCW. Are we really aware? And it's being delivered by Dr. Kaushik Nag, who is the Assistant Professor, Community Medicine, Tripura Medical College and Hospital, and Dr. BRAM Teaching Hospital. And the third speaker is Dr. Jugal Kishore. He will talk on immunization in elderly. And he is medical educator and public health expert. His qualification is MD in Community Medicine, FAMS, FIMSA, FIM, and first I
Sir, will the first speaker start?
Yes, you start with Dr. Mohammad Shahidul Rahman. You start. I already called.
Thank you, respected chairperson. Am I audible?
শুরু করো মোহাম্মদ শহীদুল,
Yes,
শুরু করো।
loud and clear.
শুরু করো, ভালো করে বলো।
Okay. Thank you, respected chairperson Ujjwal sir. Hello, everyone. Good evening. I am going to give my presentation on immunization of adolescents. Before going to the main presentation, I am to share EPI scenario of Bangladesh. EPI launched in Bangladesh in 1979. Tetanus toxoid 5 doses for women and childbearing age started in 1993. Hepatitis B vaccine introduced in 2003. Pentavalent, including DPT, Haemophilus influenza type B, hepatitis B introduced in 2009. WHO certified Bangladesh as polio-free on 27th March 2014. Pneumococcal conjugate vaccine introduced in 2015 and also inactivated polio vaccine introduced in this year. Human papillomavirus vaccine introduced in 2016. Bangladesh achieved Gavi Vaccine Hero Award in 2019 for its tremendous effort for vaccination in below 1 year of age child. Immunization coverage in Bangladesh below 1 year age child is 82.3%. So if we look in young children vaccination, coverage is excellent, whereas in case of adolescent immunization, it is grossly neglected. It's also, there is waning of immunity of childhood vaccination as vaccine-induced antibody level decline over time. There are catch-up immunization for adolescents born after the immunization introduction of newer vaccine. Why we vaccinate adolescents? To protect them against the disease that have higher morbidity. As we know that hepatitis A and varicella are less severe in young child, but it may cause severe disease in case of adolescent and adult, and or higher incidence of mumps meningococcal infection during adolescent period. Boosting of the waning immune response of certain vaccines administered during infancy or early childhood, which includes measles, pertussis, tetanus, or diphtheria. Protection against cervical cancer, which is the most common cause, one of the most common cause of death worldwide, adulthood childbearing women caused by human papillomavirus. Vaccine preventable disease control or elimination project like measles elimination, congenital rubella syndrome control program. High risk behavior in adolescents as they sometimes substance abuse, IV drug abuse, or unprotected sex exposure can increase the risk of vaccine preventable disease like hepatitis B and human papillomavirus infection. There's some mandatory vaccine for travel and abortive study like meningococcal vaccine. We can give adolescent vaccine as a catch-up vaccination, which I previously mentioned. Also, we can give a vaccine for high risk group of which like asthma patient. long-term steroid therapy, yearly influenza vaccine. There are some special circumstances like dog bite, we can give anti-rabies vaccine, or travel to an endemic area, we can give Japanese encephalitis vaccine. So if we divide adolescent, see that adolescent immunization can be divided into mandatory vaccination, catch-up vaccination, and vaccination in special circumstances. Mandatory adolescent vaccination includes Tdap or TD, which starts at 10 years of age, human papillomavirus vaccine, 9 years of age, and Bangladesh gives also COVID-19 vaccine over 12 years of age. So first of all, TD or Tdap, which I previously mentioned, that is recommended age is 10 years, only one dose followed by TD every 10 years. Tdap is preferred over TD because it contains also pertussis. And minimum age for Tdap is over 7 years. Below 7 years, it was not given. And the difference between Tdap and DTP is that in DTP vaccine, it contains full strength tetanus, diphtheria, and pertussis. But in case of Tdap, it contains full strength of tetanus, but 1/5 strength of diphtheria and acellular pertussis. In case of pregnancy, we must give tetanus Tdap during second trimester, and it is recommended in every pregnancy. And CDC recommended of giving Tdap in 27 to 36 weeks of pregnancy. And cooking strategy, vaccinating all contacts with Tdap who are over 7 years old, who might come in contact to newborn, to form a protective ring of immunity from this disease for infants. What we should do in case of unvaccinated individuals. WHO recommends TD combination vaccine for unvaccinated individuals over 7 years old. In Bangladesh, we administered first dose TD at 15 years old, second dose at 1 month apart of first dose of TD, third dose after 6 months of TD, fourth dose at least 1 year after the third dose, and there is a booster dose which was given at least 1 year after the fourth dose of TD. Pertussis wholesale vaccine is not recommended for adolescents. As we know, pertussis wholesale vaccine is given in infancy in Bangladesh. Human papilloma virus is causes actually, it's one of the most common cause of cervical cancer in throughout the world. There are 33,000 new cervical cancer case every year, and with 5,000 deaths every year. There are more than 100 serotypes of human papillomavirus. Among these, 13 to 15 serotypes are oncogenic. Type 16 and 18 are responsible for more than 70% cervical cancer. Type 16 and 18 also responsible for cancer of the vagina, vulva, anus, penis, and oral cavity, but it's less occurred. And there's type 6 and 11 are associated with anal and genital warts. Human papillomavirus vaccine recommended age is 9 years. As in 9 years, the immunogenicity is actually better. So before exposure to any sexual activity, we must give in 9 years. And if we give human papillomavirus in 9 years, then the dose is actually 2. But if we give the vaccine from 14 years or above, then there is 3 doses is required. There are worldwide 2 known vaccines of human papilloma vaccine are available. One is bivalent human papilloma vaccine, which is known as Cervarix, and quadrivalent human papilloma vaccine known as Gardasil. Human papillomavirus bivalent contains type 16 and 18, and whereas Gardasil contains serotype type 16, 18, 6, and 11 also. But efficacy, if we see the efficacy, both bivalent and quadrivalent vaccine have more than 90% efficacy for the reduction of cervical endothelial neoplasia and adenocarcinoma in situ. COVID-19 vaccine must be given in 2 doses for above 12 years and 18 years, and a booster dose is recommended. Regarding catch-up immunization, we used MMR vaccine, hepatitis B vaccine, hepatitis A vaccine, typhoid conjugate vaccine, varicella vaccine. MMR vaccine is the aim to prevent congenital rubella syndrome, recommended for all adolescents, especially girls, irrespective of their previous immunization status, and there is no upper age limit. 2 doses at 4 to 8 weeks interval. As it is a live attenuated vaccine, it must avoid during pregnancy. Actually, in previous session, we already know that hepatitis B vaccine is the universal vaccination. Previously, unvaccinated adolescent must be given 0, 1, and 6 months, and there is no booster. Hepatitis A vaccination, 85% of the below 2 years old child and 50% of 2 to 5 years old child remain asymptomatic, which are infected with hepatitis A virus. But the adults are symptomatic in 70 to 95% cases with a mortality of 1%. Hepatitis A virus can lead to acute liver failure. There are 2 types of vaccine, hepatitis A vaccine. One is killed vaccine and one is live vaccine. If we give killed vaccine, there are 2 doses, 6 months apart from 1 year onward, and if live vaccine, it's a single dose from 1 year onward. If a post-exposure person is exposed to a hepatitis A patient, then he must be vaccinated within 10 days of exposure. So from previous session, we already known that typhoid is one of the most common cause of mortality and morbidity among communicable disease, and it is the most common cause, one of the most common cause of communicable disease in this Southeast Asia region, and it is common in school-going age, because school-going age people's children are frequently take street foods. And in Southeast Asia region, 50% or more of the strain has multiple drug-resistant strain of typhoid. Typhoid conjugate vaccine single dose from 6 months to 45 years is needed. Typhoid polysaccharide vaccine is less immunogenic and multiple doses are required. Varicella, that's chickenpox, is less severe in case of young children, but it's adolescent and adult, it's develops severe disease. So we must give 2 doses of varicella vaccine 4 to 8 weeks apart. And in case of post-exposure, we must give vaccine within 72 hours. Must be avoided in case of pregnancy, and if adolescent girl receive this vaccine, she cannot be pregnant or conceive within 4 weeks after vaccination. There are special circumstances. We give some vaccines like influenza vaccine, Japanese encephalitis vaccine, pneumococcal vaccine, meningococcal vaccine, rabies vaccine, and yellow fever vaccine. Influenza vaccine, 1 dose every year if patient is immunocompromised, and it is a mandatory vaccine in CDC, which they actually immunize their child, adolescent, every yearly by influenza vaccine. And age from 6 months to 65 years, and first vaccination if the child is below 9 years old, 2 doses is required, 1 month apart, and if it's more than 9 years old adolescent, then only single dose is required. Japanese encephalitis vaccine is needed for the travelers planning for a long stay more than 1 month in endemic regions during monsoon or post-monsoon months, where it's especially in Karnataka, I think, and Tamil Nadu, the endemic regions. And doses are 2 doses at 4 weeks interval. Pneumococcal conjugate, pneumococcal vaccines are actually 2 types, pneumococcal conjugate vaccine and pneumococcal polysaccharide vaccine 23. Both are used in certain high-risk group children like HIV infected children, asplenia, that is functionally, anatomically, or surgically removal of spleen after nephrotic syndrome, sickle cell anemia. Pneumococcal polysaccharide vaccine 23 not recommended before 2 years of age, and we give a single dose. Single revaccination with pneumococcal polysaccharide vaccine 23 administered must be after 5 years. Administration of polysaccharide 23 at least 8 weeks after the last dose of PCV vaccine with underlying medical condition, including cochlear implant. Meningococcal vaccine, actually in this subcontinent, meningococcal A is more common. As we know, meningococcal can actually causes severe meningococcal septicemia and one of the cause of death actually. Recommended for only for during outbreak, immunocompromised person or students going to study for abroad like USA, Canada, UK, travelers for Hajj or Sub-Saharan Africa, single dose of quadrivalent conjugate vaccine containing serotype A, C, Y, and W in individuals 11 to 15 years. Booster dose is required in persons with complement deficiency. Oral cholera vaccine is required in case of endemic area with outbreaks, 2 doses, 1 to 2 weeks apart. And yellow fever vaccine is a single compulsory dose for all travelers above 6 months of age traveling to endemic zone like Sub-Saharan Africa or tropical South America 10 days before the travel. Following yellow fever vaccination, the certificate is valid up to 10 years. Barriers of adolescent immunization is the lack of awareness, both in our side and also in public side. Providers, personal bias, like we are less talk with our patients about adolescent immunization. Negative media reports, sometimes media covers negatively of adverse immunization reaction. So people are not interested to vaccination. Lack of past reports that if a vaccine give to adolescent and its effect is good, like that reports are not so actually common in our country. So we can't encourage people for adolescent immunization. There is some fear like sore arm or vaccine phobia also happened to some people. So vaccine actually not given by them. And as we less prescribe adolescent vaccine, there's poor stocking of adolescent vaccine. So how can we overcome these barriers? By social media awareness, send reminder messages at their after 10th birthday, awareness events at clinics, school, colleges, NGO, social clubs, vaccination program at school and college. Mahatma Gandhi says, the future depends on what you do today. So that's all from my part of presentation. Thank you all.
Thank you, Dr. Mohammad Shaidur Rahman. The question answer session will be taken later on. Now I request Dr. Kaushik Nag, Assistant Professor, Community Medicine, Tripura Medical College and Dr. BRAM Teaching Hospital. And he will speak on immunization for HCW, healthcare worker. Are we really aware? Dr. Kaushik Nag, please. Dr. Kaushik Nag.
Good evening. Good evening. Good evening, all.
Kaushik, bolo.
I need to share the screen, I think. I have shared my presentation? Can from the host site they can present my PPT?
Kaushik, you can request the host to share the screen from his end. Yes, yes. It's visible now.
Yeah.
Carry on.
Thank you. Yeah. Thank you. So, good evening, all. I am Kaushik Nag, and today's topic is immunization for healthcare professional. Are we really aware? So, this is a very important topic. Can you go to the next slide, please? So the content of my presentation will be background, vaccine for healthcare professional, and what are the barriers for vaccination and strategies to improve vaccination. Next. So to start with, we healthcare professionals are at, as we all know, increased risk of acquisition and transmission of infectious disease. And different studies in India have shown that we have very low vaccination coverage for influenza and varicella. That is as low as 4.4% for influenza and 16% for varicella. And there are several barriers to vaccination uptake in healthcare professionals. Though we know what is the vaccine to take, but sometimes some barriers are there. I'll discuss later. And vaccinating healthcare professionals is an essential preventive measure to protect them and their patients. Next slide, please. So these are the vaccines commonly we can prescribe for us: hepatitis A, hepatitis B, MMR, meningococcal vaccine, influenza, pneumococcal, varicella, tetanus, diphtheria, pertussis, polio, typhoid, rabies, COVID-19. So I'll discuss one by one, and I'll try to say different guidelines. What is the guideline saying according to World Health Organization, CDC, or say API and other Indian Medical Association. I'll show in my subsequent slides. Next slide, please. So regarding hepatitis A, as already our expert Dr. Anirban has already mentioned, for adults or like healthcare professionals, as API, WHO, and CDC or Advisory Committee on Immunization Practice have not recommended such, but Indian Association of Occupational Health has given some recommendation like single antigen vaccine. We can give 2 doses, 1 ml IM at 0 and 6 to 12 months. Then if we use combination of hepatitis A and B, then we can give 3 doses like hepatitis B vaccine, that is at 0, 1 and 6 months. And IMA has recommended about 2 doses at 0, 6 months for inactivated, and for if it is alive, then 1 dose. So this is not so important. Next, hepatitis B, as already discussed elaborately, I just show you the guidelines. Next. So regarding hepatitis B, we have guidelines, and we know that it will be given as 0, 1, 6 months, 1 ml intramuscular, according to API, IMA, and Indian Association of Occupational Health. WHO has said that unvaccinated healthcare professionals who may be exposed to blood and blood products work, they may be, they should, or they must have vaccination against hepatitis B. And CDC had told that unvaccinated or those healthcare professionals who has incomplete series or no vaccination history or no up-to-date immunization status, they can have 2 doses at 0 and 1 month. Otherwise, they can have 3 doses at 0, 1 and 5 months after the dose 2. I'm not going in details because it was thoroughly covered in the earlier presentation. Next. Regarding MMR, API has told about 2 MMR doses at 4 to 8 weeks interval. Same IMA and Indian Association of Occupational Health also recommended. And if it is the healthcare professional previously immunized, then single dose also sufficient. According to WHO, they have told that regarding if there is any proof or documentation of immunity or immunization should be required as a condition of enrollment into training and employment. And they also say healthcare professionals are not at risk of mumps, but I don't know, in our country there are lots of mumps also cases are there in healthcare profession. Then CDC told that those persons who are born in 1957 or later with no MMR vaccination, or those who don't have any up-to-date immunization status, they can have 2 doses of MMR with a gap of 28 days. Next slide, please. Regarding meningococcal, API has told about a single dose during an outbreak. WHO told about 1 booster dose, and that may be 3 to 5 years after the primary dose may be given to person considered to be at continued risk of exposure, especially the microbiologist, for those who are dealing with this organism most commonly. Then CDC told about same 1 dose for those who are routinely exposed to Isolation Neisseria. And IP and IMA also told about 1 dose and booster every 5 years if the person is still at the high risk. So this is about meningococcal vaccine. Next. Regarding influenza, as we all know that we need annual shot, that is 1 dose every year. But API has no recommendation for that. But other organizations like WHO, CDC, IMA, everybody, and IMA also has said about importance of influenza vaccine. And if it is trivalent inactivated, then you can give single IM or intradermal every year, or if it is live attenuated, you can give intranasal. This is also a very important vaccine because influenza cases are also very high in our country and also in different countries. So the healthcare professionals must have this vaccine, influenza. Next slide. Regarding pneumococcal, there is no as such recommendation for healthcare professional. But yeah, if the person is elderly and if any comorbidity that is more, if the person is having chronic kidney disease, chronic liver disease, diabetes, or the person is elderly 65, more than 65 years, we can give PPS, that is polysaccharide, pneumococcal polysaccharide vaccine. And conjugate is mainly given for the infant that is less than 1 year. And it was also discussed in earlier presentation thoroughly. So I'm just focusing on the guidelines, whatever, because most of the things we know that these are the vaccines available, but sometimes due to some reasons we don't take the shot, or I'll tell about the barriers later. Next. Regarding varicella, API has recommended that if it is a live attenuated, then we can give 2 doses, 0.5 ml subcutaneous over delta at a gap of 4 to 8 weeks. And WHO has said potentially susceptible health workers, that is unvaccinated or with no history of varicella, they can have 2 doses. Same is recommendation by IMA and Indian Association of Occupational Health. But CDC told if there is no history of varicella infection or vaccination or unknown immune status, so all the guidelines is told about 2 doses of varicella vaccination. Next, please. Next slide, please. No, previous. Yeah, okay. So regarding this is also important vaccine, tetanus diphtheria. So according to API, they told about you can have 1 dose of Tdap, tetanus diphtheria and cellular pertussis if not received Tdap vaccine before, and if there is a gap of more than equals to 2 years in the last TD vaccine, and we can have a shot of a TD booster every 10 years. And as such, WHO has not given any recommendation, but they have the vaccination of healthcare professionals with diphtheria boosters every 10 years, especially those who may have occupational exposure. We can have CDC, IMA, and Indian Association of Occupational have the same guideline, that is 1 dose of Tdap, and then TD booster can be given every 10 years. So this is the guideline regarding tetanus and diphtheria. Next, please. Regarding pertussis, as such, there is no guideline in API, IMA, or Indian Association of Occupational Health. But WHO told that healthcare professionals should be a prioritized population, and CDC told that pregnant healthcare professional needs Tdap in each pregnancy, which in earlier presentation the speaker also told about it. Next, please. Regarding polio, WHO told that all healthcare professionals should have a completed primary dose, and IMA and Indian Association of Occupational Health told regarding 1 or 2 doses if the person is unvaccinated. 0. Excuse me. 0 and 4 to 8 weeks. But API and CDC has no recommendation for this polio for healthcare professionals. Next, please. Typhoid is already discussed elaboratively. So API has told about if there is any outbreak in the community, we can give live oral typhoid 21A vaccine 3 doses for the entire community, and booster we can give every 3 years. WHO has not mentioned about it, and CDC told microbiologists and others who work frequently with salmonella typhi might have a shot, and Indian Association of Occupational Health told about a single subcut or IM intramuscular dose of 0.5 ml, same as Indian Medical Association. And if they are giving unconjugated vaccine, then you can give every 3 years. And it was also discussed in earlier present. So most of the things are repetition. So I'm just focusing on the guidelines, whatever the guidelines are saying we should have, that is healthcare professionals should have. Next, please. Rabies, I think there was an entire session last day, and so I'm not going into again. Just these are the guidelines which we have IMA Indian Association. Some guidelines say 4 doses, some guidelines say 3 doses, and we have different regime, geriatric regime, medicine regime. We all know what are the things we should do post-exposure, prophylaxis, and pre-exposure also. So I am also skipping that part. Next slide, please. COVID also, it was elaborately discussed today. So as such, IMA and API has no guideline, but as we all know that we must, we all have, I think, taken the COVID vaccine, and as Chandra sir also told about booster, and other experts also told about the second booster or precautionary dose, which we might take, but the natural, if we have natural COVID infection after 2 shots of COVID, that is also sufficient. But as such, there is no guideline by IMA, FPI, or Indian Association of Occupational Health. Only WHO and CDC told that healthcare professionals should be a prioritized population, and we know that we are. I hope that all of you has taken this vaccine, and so I have nothing to discuss more on this. Next, please. This is the most important slide for me. That is the barriers to vaccination for healthcare professionals. So I have given 3 country scenarios. One is for us, India, then another European countries, and for China. So for us, the reasons or barriers, maybe we are too busy. Maybe that is not. Maybe we are last 2 years are very busy. But yeah, whatever maybe the reason, but we must have this vaccine whichever is required and recommended by different bodies. Next is a barrier, maybe not aware of vaccines or lack of knowledge, which I don't know or don't believe that they don't have the knowledge, or they are not aware, but sometimes it may be there in some rural parts of our country that somebody might know all the things. Then vaccines may not be available or freely available. There may be doubts of vaccine efficacy or safety. There may be fear of side effects. We also had lots of doubts about the COVID vaccine when it was introduced, and we had a fear what are the side effects, what is the AFI, that is adverse effect following immunization. Needle phobia, yeah, maybe one barrier. Then cost of vaccine, because these are not supplied free of cost. There's all the vaccine we have to take on own, and that may be another barrier. Then there may be low perception of susceptibility, risk of contracting the disease. And last is lack of understanding of disease or infection. So these are the barriers which may be relevant for our country. And for European regions, maybe they are already immune. Then say inconvenient vaccination timing sites, fear of side effects, adjuvants or ingredients. Then there may be same concerns about newer vaccine like COVID. Then they maybe have some doubts about the vaccine efficacy and effectiveness, and perceived low risk of contracting disease. So these are the barriers maybe we can consider for European countries. And for China, yeah, same opt-in, they might have opt-in immunity from work or natural immunity, or hospitals are not providing, too busy, or officially there is no recommendation to take the vaccine. So these are the barriers we have to look for, and we can, if we can motivate. It's all about motivation, then awareness, because awareness is most of the healthcare professional has about the vaccine, they know, but sometimes due to lack of time, due to lack of motivation, they may be not taking the vaccine. So next is how to improve the vaccination. So next slide, please. Next slide, please. So how we can improve the vaccination among us, that is healthcare professionals. One is we can do regular monitoring. It can be done by the hospital. It can be done by some bodies, local bodies, state bodies. Then we can have some incentives for vaccination. Then intensified advertising campaign. We can go for CME type, CME continuous medical education for healthcare professional. Then there may be some mandatory vaccination policy, except when it is contraindicated. Maybe regarding travel also, like which we required for our travel for COVID vaccine. for inter-district, inter-state travel, especially by air, by rail, we needed this COVID vaccination certificate. So we can do for other vaccines also, which are very prevalent and causing mortality, morbidity in our country. We can work on it. Or another maybe convenient access to vaccination and work. Sometimes vaccines are not maybe available. It may be available in the urban areas, but in rural areas, maybe these vaccines are not available because these are costly vaccines. And so that thing also we can improve. And for the UK, or we can give vaccine at their convenient time, because we have to think about their timings or schedules, what are the schedule they are doing in their workplace, so we can look and make it convenient for them. Then introduce peer vaccination and also employment conditional upon receipt of vaccine. So we can make this eligibility criteria also, that if you are not having this vaccine, then you will not be eligible to go for a job. So these are the strategies we can look upon to improve the vaccination among healthcare professionals. And with this, I want to thank. Next slide. Next. So I want to say that save healthcare professional to save world, because we are the person who will who are the frontline warriors or COVID or whatever they have told us, saying like that. So first of all, we have to save us so that we can serve better. And if we are not healthy, if we are not at our full best 100%, then how can we do our job? So let us vaccinate, and I hope we are all aware of the vaccines, whatever we take. Just it's a motivation, or sometimes maybe a push from a colleague, a friend, or family, whoever in the same profession, they can motivate each. And if they are motivated, I think most of the vaccines, whatever is recommended, the healthcare profession takes. Thank you, and I also want to thank the organizers, especially Dr. Polysaccharide, Dr. Parthakumar sir, and Dr. Shatabdi madam, for inviting me and giving me the chance to speak for this important topic. And I wish that we all be vaccinated and stay protected. And I also want to thank our chairperson, Dr. Ujjwal Sengupta sir. And I next want to hand over to him. Thank you.
Dr. Jugal Kishore, please.
Thank you very much, sir, Dr. Ujjwal. And I'm grateful to Dr. Saurabh Kole, who has invited me. I just want to know my slides are visible or not. This is the first slide. Can anybody tell me that this is visible?
Visible, sir. Yes, sir, visible.
And second one also?
Yes, sir.
Okay, fine. So my job is already done by previous speakers, because they have discussed about various vaccinations. I just want to say, when we talk about elderly, then as the life expectancy is going up and the number of people are increasing in older bracket, we have evidence from all over the world, including India, that the number of elderlies are increasing, and these are the people who are dying. Ultimately, this is the truth of life, that some day we are going to die. But the problem is that if we can increase a little bit of our age, even in elderly age group, then it is always good. So this vaccination is mainly for prevention of death and prevention of diseases. And and maximum people are dying in older, and the severity of infections are also common in elderly. Therefore, focus should be on, the group, because in public health, remember, we have two major issues. One is vaccination for the individual and vaccination for the community. And unfortunately, most of the study done at the individual level, and we don't have community trials to say that if we have the national program or if the policy is made to adopt the vaccination, what would be the effect on the population. So we require a lot of studies in this area, which is almost not there. We already know about the COVID pandemic, and these are the people who have died more, elderly. And, infectious disease, long-term sequelae of the infectious disease leading to disability, impairment in the daily living, and frailty is also change, more there. The loss of independence is there when we have this problem. So there should be focus on elderly, and this is the most neglected area and neglected age group also by all. The eradication of smallpox and polio elimination has made the success that at the community level we can eliminate or eradicate majority of the infection if we have very good program, if we wish to, and that is also the development of science, that in COVID we have able to develop the vaccine in a very short period. So the maximum effect should go to the people, and the people are actually those who are suffering more. So that is why I'm saying the elderly is the most important group. Now I'll just go through fast because I know it's I'm the last one, and one important speaker is there after me as a general, then I should not consume time. But these are the recommended vaccination for elderly, particularly the pneumococcal disease, which is quite common world over, and community acquired pneumonia is very common of cause of death in elderly. Therefore, we have to have the vaccination, and we have the vaccine, and we have the polysaccharide vaccine, and it induces a very good immunity and antibodies are increased. So pneumococcal is given and should be made available wherever is possible in those areas, particularly in old age home and where the elderly are more commonly found. So this 7-valent and 10-valents, and this is already going on, and now we have 13-valent vaccine. So in future, maybe I think we'll have very good vaccination for elderly, particularly for pneumococcal. Another one is a herpes zoster. I have seen cases where this infection which occur in childhood, and later on you develop the herpes, and it's a very, very painful condition and leading to side effect, which is, or maybe complication, which is very, very troublesome. So vaccination may prevent these all things. So this is another one where we can reduce the post-herpetic neuralgia and other things which can debilitate the elderly person. Then we have another vaccine. So herpes zoster is well studied and shown very good effect when we give to the elderly, and antibodies response are quite good when we give second dose. So another one is the influenza vaccine, which is after the H1N1, and this is keep occurring every year, and people are dying, particularly the elderly. So quadrivalent vaccine is available, the trivalent and quadrivalent in some places. Even in government setup, we are advocating that this can be given to the doctors. He the Kaushik has already said about healthcare workers and occupational group, but this influenza vaccine is, should be part of it beside the COVID and other. So the challenges are many, but I think meta-analysis showing the benefit of influenza vaccine and should be given. Another one is, I'm not going into the detail of, how much should be given, and these are all there, and many studies are showing, I've given the references, so the studies are there, but they are actually on very small group, and risk reduction is also not to that level, which we can say that it should be the policy, community policy. Other vaccine which is there, like the tetanus, which is preventable and can be given every 10 years, which can give. And the idea of vaccinating with tetanus after every injury is not a good idea. So therefore, if we have continuous vaccination, so there is no need of on every, injury. Diphtheria is a, interesting vaccine, and it keep occurring, and now in older population also, now we are seeing cases. So there is no harm in giving the, the tetanus and diphtheria together. Maybe sometime pertussis. Polio is already, eliminated from India, but children are given. But in some cases, when it is required to some elderly people, can be given. So beside that, we have the typhoid. We have discussed about typhoid, hepatitis A, and cholera vaccine in certain, areas or certain group. The risk is high, and typhoid particularly is very serious condition. Although when you age, the occurrence of typhoid is less as compared to younger population, but still, if we can protect when we go in certain areas where infection can be there, so you can decrease. So older vaccination, you can see that different vaccinations are there in different countries, and they are given above 65. In our country, it is, more than 60. So we can adopt some of the vaccines which can easily be given at the individual level, but at the national level, we require studies to show that yes, they are important. So tetanus, diphtheria, they are all appear to be useful. Pertussis is also very useful. So hepatitis B, we have discussed about that. My previous speaker has already said that how hepatitis B is preventing, although it is part of our national program, but a booster dose can be given in adult and also in elderly, which can definitely decrease the chance of our infection, which is quite dangerous. So the new vaccines are coming up with the new molecules and new techniques. So they should be, advocated. But as a public health person, I am always looking forward for community trials where we can adopt the vaccine for the national program, or we can include in the national program. There are studies showing that universal immunization with the influenza vaccine have a much better effect and a better compliance. So such technique, such type of studies are good. Similarly, pneumococcal universal vaccination has shown a better effect than, just, isolated cases of vaccination. So these two areas, I found that yes, some type of vaccination trials are required at the community level. Then only I think the government would take serious note of it, particularly for older person. So in the end, I just want to say that these are the group which are, at high risk of infectious disease, and vaccination can definitely prevent. So we can make the healthy aging. That is why it is a very strong case. We already have the childhood vaccination. So if we can continue in adult and elderly, I think we can make the universal vaccination very easy. Then it should not be that, the vaccination only for children. So that should not be the idea. It is a myth. Similarly, we have, various diseases which have proven that it can be done. So regional difference can be there. We require epidemiological data for this, but definitely, the program like this can pull all the data together and can have very good, policy decision that what should be done and the government should do what. So that is what I wanted to say. So thank you very much. This is what I want to say. Thank you. I hope I am not taking much time. The doctor as well.
Thank you, Dr. Jugal Kishore. And now I hand over to the moderator, please. This is very important topics covered by you and all the speakers. Is there any question? If there is no question, we may pass on to the next session.
Yes, sir. Sir, this was a mesmerizing session. Actually, all the speakers, Dr. Saidu Rahman, Dr. Kaushik Nag, and of course our Professor Jugal Kishore sir, enriched us so much.
They gave no scope for any question.
Yes,
that is the main thing.
Yes,
so excellent discourse. You may pass on to the next session. Already we are running late. Okay, thank you.
Thank you. We are concluding this session.
Thank you.
Please join the session. That is the deliberation from Dr. Reda Satki. He is the founder president of Geneva Learning Foundation, World Health Organization. All of the speakers and chairperson are requested please to join there.
All right, thank you very much, Dr. Mitra, distinguished speakers and leaders of immunization in India and everywhere else, given this is a World Congress on Adult Immunization. I'm going to go perhaps very quickly, and this presentation may not look like a conventional presentation suitable for a World Congress. Some of you may even be surprised or I hope not offended, but and one of the ways in which it is going to be different is that you'll be hearing music and perhaps other sounds as we go. So first of all, I'd like to say this is very timely. I think there's a small technical glitch, as always happens, of course. This is very timely because Immunization Agenda 2030, so the strategy adopted by all of the member states of the World Health Organization, as you know, places as one of the key strategic priority for life course and integration, with the goal that all people benefit from recommended immunizations throughout the life course, effectively integrated with other essential services. However, the story I'm going to tell you will show that it's not so easy. And in fact, recently I had a chat with someone from the headquarters of the World Health Organization, one of the people responsible at the global level for life course and integration, who said, what can we do to get more interest from professionals who work in immunization in this question? So that is one of the ways, one of the questions that I'll be trying to answer. Now, our work since the 29th of April, so World Immunization Week 2021, has focused on this question, really thinking about the future of immunization. And at least for this decade, it's focused on Immunization Agenda 2030. Now, just if you can easily, if you don't get everything you want from this presentation, if you'd like to know more, just look up the Geneva Learning Foundation or my name, and I welcome, happy to connect with you and encourage you to connect with the foundation. Another thing is, one of the things we're experimenting with, and this is part of our mission to find new ways to learn and lead, is really to, we're now offering a podcast, and I encourage you to go look at that if you'd like to know more about our work. Now, very brief introduction to the foundation's work. We develop a number of programs. It's about transforming how we learn and lead. The basic change in the model is shifting from old learning, where you have a teacher at the center addressing the students, transmitting information to the students, going to a new learning model in which teachers are learners and learners are teachers, connected to each other around this hub and spokes where everyone is connecting with everyone else. Here at the right, you see one of the main theoretical models and frameworks that we rely on from Bill Cope and Mary Colensis, which is really an agenda for new learning and assessment. What that means in practice, for example, in our partnership with the World Health Organization, we've developed already, I think to this day, we're now at the 30th, we're currently running the 30th cohort. The last 4 or 5 cohorts have been specifically on Immunization Agenda 2030. And one course in particular really helped immunization professionals look at this question of life course and integration. Now, with the first evaluation in January 2019, this is what we found. These are more than just courses. These are interventions designed to foster and improve practice at every level. Together with, in fact, Dr. Mitra, other scholars from India and 56 other countries, we then built what we call the Impact Accelerator, a new kind of system to help immunization professionals tackle difficult questions and lead change, including this change from where we are now with vaccination focused primarily on small children to a more complex but also more valuable and more productive immunization system that looks at immunization all along the life course. We found that this system, for example, within 6 months could produce startling results where we saw that people who engaged through this impact accelerator were in fact able to lead change far more quickly, with almost an 8x difference. We're 8 times more likely to actually develop a meaningful, tangible project around their needs, their challenges at the local level, wherever they work, and actually take that project forward. So where we are today is, as of this morning, our platform connects 43,981 immunization professionals. And again, in this decentralized model, it is not only about what we, as the Geneva Learning Foundation, transmit to practitioners, it is also very much about practitioners sharing with each other. On the 29th of April 2021, you can see here the 2 top global people from UNICEF and WHO respectively, Dr. Robert Nandy and Dr. Anne Lindstrand, who met with over 5,000 immunization professionals in an online event from basically all over the world. And they were there to present Immunization Agenda 2030, but they were also there to listen to the challenges. And I would like to say that life course and integration was cited as one of the top challenges, but it was not. And I think this is one of the many reasons why I see this particular event, the World Congress that you are organizing starting today or kicking off starting today, as a key event, because right now the perspective of life course and integration is not at the top of the agenda. It is very prominent in SP4 of Immunization Agenda 2030, but I believe, my conviction is that it is the movement, the mobilization of professionals like yourselves who will be in the coming weeks, months, and years making the difference, putting this question at the heart of everything that happens in immunization. This question of how we lead change in immunization, for a long time, maybe we did not really need to ask it. Immunization was one of the few interventions in global development that actually worked, where we could line up the kids in the village and they stopped dying stupid deaths from diseases that no one should be affected by, much less die from. Now we have a more complex environment of obviously a changing world, and therefore it is necessary. We feel that there is an imperative to move again, to mobilize again around questions that are new, to advocate for change in respectful, constructive ways, but change is certainly needed. So on the 7th of March, and you can see here so the sort of slogan and banner, this call to join the movement for immunization. This is not, a kind of just activist or rabble-rouser movement. This is a movement called for by the Director General of the World Health Organization at the 74th World Assembly, calling for a broad social movement for immunization that will ensure that immunization remains high on global and regional health agendas and help to generate a groundswell of support or social movement for immunization. Now, this movement is larger than any one individual or organization, but I believe an event like this World Congress is positioned and uniquely positioned to contribute to that specific question, a strategic priority of Immunization Agenda 2030, that despite the recognition of science that it is an important question has not yet been translated into policy practice. That is why we need initiatives like yours in order to help build and propel this movement, and to make sure that all of the questions that matter, all of the questions that can make a difference, can actually just be part of this movement for change, to make sure we can achieve the global goals of Immunization Agenda 2030. What's at stake? The estimate by the World Health Organization is that achieving the goals, including strategic priority 4, life course and integration, will save 50 million lives. That is the estimate from WHO. So again, through this World Congress, you are contributing to a critical question that is part of this broader movement to save lives. Now, at the Geneva Learning Foundation, our contribution, we look at education, we come through the door of education, and we've been working across a number of areas, across medicine and public health, as well as humanitarian response. We've developed an approach called the full learning cycle, and on the 7th of March, our contribution to the movement was to call for immunization professionals to join what we call a full learning cycle. 6,319 professionals responded to this call, and of those, 151 were from India. You can see the list here of all the countries involved. You can see that still many countries have one sole representative. India is number 4 in the ranking. It is not a ranking, but it is a reflection of the extent to which there were immunization professionals at that point in time who were already ready to respond to the call. The call is still very much open, even though the movement has now been launched, as we progress through this continuum of action from knowledge to impact. Now, you may ask, what is this full learning cycle? What is it about? What is the curriculum? What is the content? Well, we flipped on its head the logic of content being pushed from Geneva or Washington or London or Seattle to people in countries. And instead, we are asking you the question. You work in immunization every day, whether you are in a health facility, whether you are in a district, a region, or working at the central level with the Ministry of Health. And so we asked you, what are the challenges that matter to you? And every person who applied to join the movement responded to over 95 questions, including the questions to identify their biggest challenges. And again, life course and integration, adult immunization, was not in the top reasons, despite its recognized importance. Right now, what this gives us is a picture of how much work remains to be done, and therefore how important events and initiatives such as yours are to the recognition of this strategic priority, that this is indeed a strategic priority. Now, in the full learning cycle, we have launched what we call the Ideas Engine. I'm going to very quickly tell you more about that and invite you to go see for yourself. And of course, call on you to, if you find this compelling, if you see this as a potentially useful way to strengthen the work that you are doing, to join this movement for Immunization Agenda 2030. The Geneva Learning Foundation will be offering 3 levels of certification for the different components of the full learning cycle, and there is very much a modular approach where every participant decides where, when, and how, and where they want to engage in order to gain the greatest benefit that will help them, that will help you in your daily work. Dr. Godwin Mindra from UNICEF made the point that in this, even though there are people from all levels of the health system, we are at a juncture where the difference is going to be made. The point of contact, where the rubber hits the road, will be where those of you who have direct contact with the communities are going to use new ways to do new things, and that is all about reaching the unreached. Kate O'Brien, the director of immunization at WHO, we need people to feel like they have the authority and are empowered to lead change in their community, in their program, at the most local level, understanding what the goal is and what the targets are. And that means that if someone who is working at one of the so-called lower levels recognizes the significance and importance of adult education, they should be adult vaccination, they should be supported. They should be that initiative, the willingness to integrate adult vaccination into the ongoing program historically focused on children needs to be recognized and supported rather than shunned or seen as something that threatens the existing way that things are done. Now, what does this movement mean in practice and what has it generated? We've seen incredible levels of motivation, excitement, and enthusiasm. And this is from Asad Ali, who is in Pakistan. And even though he is obviously very busy, he takes time to actually let us know. He says, I'm in a mountainous region of Pakistan on the COVID-19 vaccination surveillance and monitoring mission. And he has the kindness to let us know when he will be back. That is how committed he is to contributing to the global movement, even as he continues to do his daily work. And this is from Afghanistan. And Syed writes and says, very simply, only one line. We asked him if he was ready to join and to contribute and to begin the activities of the full learning cycle. And you can see his response here. It is just one line, but it is quite eloquent. From an immunization professional and a doctor, he says, yes, I am ready to rock. Now, what do we actually do in this full learning cycle, and how could this be helpful to you and others who are leading the change needed to have adult immunization recognized and integrated into existing immunization programs in India and elsewhere? First example of the first component, what we started with on March the 7th, was the ideas engine. And what this is across these 7 strategic priorities of Immunization Agenda 2030. You can see there's one here, SP4. This is the one that every one of you can contribute to, and also go and look at the ideas and practices that are being shared from participants across the world. It is as simple as clicking a button, and in the first 10 days of the movement, 764 ideas and practices were submitted from 62 countries. And this came from 671 scholars, practitioners who shared these, including a small but significant number of ideas focused on SP4, on life course and integration. Now, again, the ideas and practices by country, you can see the list here, which countries contributed more. And I'm proud to say and report that India shared 27 ideas and practices, 3.5% of the total. Now, before I go any further, this idea, yes, and this is a quote from a participant. I encourage you the Ideas Engine is open to all members of the Movement for Immunization Agenda 2030. It is a public good that we offer the structure, but it is being filled by practitioners from all over the world. So if you want to see what is being done, what are the practices, what are the ideas in other parts of the world, that is what is offered through the Ideas Engine. I'd like to mention another initiative that is now part of this movement for Immunization Agenda 2030, and that is Teach to Reach. For the first time on the 17th of June, we're going to be opening up Teach to Reach, which historically was a networking event reserved for immunization professionals. And for the first time, professionals from other areas of health are going to be welcome to join. This makes it relevant because life course and integration requires a more integrated approach. We cannot continue to work in silos. And just to give you an indication of the scale on which these networking events, this Teach to Reach happens, the last Teach to Reach on 4th of March saw more than 8,500 immunization professionals from all over the world connect and support and meet and learn together. Now, what's the point? What's the value of this and other activities in the Movement for Immunization Agenda 2030? You can see one quote here. I had the privilege to interact with very highly qualified, internationally recognized health professionals. It's like an opportunity to dine with the creme de la creme of global health. And the second quote from someone who is probably closer to the ground and works in a rural area and who's been wrestling with vaccine hesitancy, and who says, after Teach to Reach Connect, I have evidence to convince any resistant people. This event was amazing to me. Of course, if you work in public health, you're asking, well, okay, what's the impact of this? Where's the evidence that this is actually making a difference? We can spend time talking to each other, connecting with each other, but so what? And so on the so what, we're using an evidence-based scale tracking, not only taking the temperature of what is happening throughout the movement with people, individuals reporting to each other and to us what difference it's making in terms of a gain in confidence. If you're going to go advocate that adult immunization needs to be a focus of your immunization program, you need to have the confidence to do it. If you are going to consider changing how the existing system that is entirely focused on children needs to expand to teenage girls, to adults at different points in the life course, you're going to need motivation. And you can, of course, rely on the ideas of colleagues who are right there, your immediate colleagues. And of course, they're the ones you work with every day. They're very important. But this idea of a global support system, imagine if all of a sudden you can benefit from the inputs, the ideas, the practices of thousands of other fellow immunization professionals from all over the world. That is the power of this approach and the potential of this movement. After participating, I thought I need to find out more technical details about my country's program. So this is someone working in the health facility who all of a sudden, rather than waiting to be told, do this, do that, may go look at the country's program, see that adult immunization is already part of the story and say, how come we're not doing it? So imagine that kind of local initiative. If all of a sudden people at every level of the health system are speaking to each other, connected to each other. And this is not a dream. This is what we're doing every day in the movement. And this is what we have done since the COVID-19 pandemic came and we created what we call the COVID-19 Peer Hub. So again, to finish, this is about the need for change and recognizing that leading change in a globalized, hyper-connected world cannot happen in isolation. We simply cannot continue to do things, learn things, and lead change in the ways that we have before. Last but not least, you can see the QR code here. If you'd like to experience this is again a testimonial from a participant. Every Monday we have an assembly. You do have to apply to join the movement. It is actually closed, but I'd like to, on an exceptional basis, we'd be honored to have members of the World Congress join us. It is every Monday, so the next one is actually happening tomorrow it is quite late Indian time because it is 5 p.m. Geneva, so I believe that would be 8:30 p.m. Indian Standard Time. Thank you very much. I think I apologize if I have been over time. I hope that Dr. Mitra will forgive me for that. All right, thank you very much, Dr. Mitra, distinguished speakers and leaders of immunization.
So thank you, Dr. Reda. So we are just going to the concluding part of our program. I will request the faculty and the delegates who are here, please stay with us for next 10 minutes. And really, it's an emerging experience, wonderful experience for us. With the help of technology, we are being able to organize this program. And but one thing I should say, the journey is just started today, and the faculties and the delegates who are here, in future, we want to work with you. You are a great strength of Indian Society for Adult Immunization, and I'm sure that in future we'll be able to work together, and definitely we'll be able to improve the scenario of adult immunization in our country. At the same time, I should say that what Dr. Reda was telling, really we'll also work with his movement, with their movement, and to improve the adult immunization scenario all over the world. I just want to say a few that some of the doctors who are working for the last 1 month, day and night, to organize this session, I just want to hear from them also. I asked Dr. Shatabdi to say a few words.
Thank you, Dr. Pole, sir, and also all the chairpersons, the speakers, and our very respected audience, the participants who were there to make this successful. And we are really overwhelmed with the presence of so many participants in the weekend especially, and during the season of the festivals for many of them. And for the, especially for the foreign speakers who are from our very close neighbor Bangladesh, from UK, from France, Malaysia, and also many, and Switzerland. We are very much thankful to all of them. We hope we will continue our journey in future. Thank you. I am requesting Dr. Arijit De to tell something.
Hello everyone. It was an extreme pleasure. It was an extreme pleasure to be a part of this movement. I will call this as a movement because this is what we started. Okay. In these 2 days, we just planted the seed, as Dr. Polysaccharide said, and we will bear the torches and keep running with it so that this does not die. And this is the need of the hour, as COVID-19 showed to everyone, especially the adults, that we are not immune. Rather, we need immunization so that we can continue with our journey. In this regard, I also must say that this program, or rather this this body, ISAI, has been the brainchild of Dr. Polysaccharide and especially Dr. Pasteur sir as well. And they have worked tirelessly for the last 10 years almost so that their efforts can bear their fruits. So as I said, this is just the beginning, and I would request all the delegates and all the participants to be associated with us, to be in touch with us, so that we can actually plan something and make something materialize. Thank to all of you, and I would like to request Dr. Debatta Halda to share her thoughts.
Thank you to all the international national faculties. A very significant moment for us today, as when all the members of ISI, Indian Society for Adult Immunization, when we first met Polysaccharide sir, this society was thinking of organizing a conference. So it was a very rapid thing. We decided on within 2 months that we are going to organize this program. And the most important part, all the faculties who have joined, all the chairpersons who have joined have supported us tremendously. Whenever we have approached them, they have replied back, they have responded. So a very thank you to all of them. We hope to work with all of you in near future and grow this society. Thank you. I would request Dr. Partho Karmakar sir to say the vote of thanks on behalf of all of us.
Thank you, Dr. Debatta Halda. So, good evening, one and all. So, vote of thanks is the last, but not the least, very important part of any conference in the valedictory session. So, we have come to an end of 2 days academic extravaganza of first ever World Congress on adult immunization. So first of all, I'm to name a person, that is Dr. Saurabh Kole, who has played the pivotal role in making this conference a success, as well as making this conference going to happen in this Immunizations Week, World Immunization Week. So many of us had some doubt that whether we will be able to organize it in this World Immunization Week, because we had very short time, but ultimately by cooperation of all, we had been able. Then next, we like to express our respect to one of the senior most, but yet most dynamic person, that is Dr. Muruganathan sir. He all the time inspired us for organizing this sort of adult immunization program to increase awareness among the doctors as well as the people from all walk of life. So we are really thankful to Dr. Muruganathan sir, and we expect his grace, his blessings, his inspirations all the time in our future work. And as already said by our Dr. Shatabdi, as well as Dr. Debatta and Dr. Arijit, that we are really very, very thankful to all the speakers and chairpersons, national and international, for sharing their knowledge, their experience with us, as well as with the learned gathering, so that our knowledge has definitely become a little bit wiser, and our future planning will be smoother through this knowledge and everything. So we are really express our heartfelt gratitude to all the speakers as well as chairpersons. Now, it is a very pleasant moment for us to congratulate as well as to express thanks from core of my heart to our active organizing committee members like Dr. Reena Ghosh, Dr. Devdatta Haldar, Dr. Shatabdi Mitra. Many times I apologize. I said her that Shatabdi Ray, but she's Shatabdi Mitra, Dr. Ribhu Basu, Dr. Arijit De, and Dr. Shivaji Gupta. Without their cooperations and their relentless work, this conference may not be that successful what it happened in last 2 days. Now, you all know, whenever we are organizing a conference in Zoom platform, technical part really plays a very important role, and it is of paramount importance. So we really got too much cooperation from 3-4 persons, and it is all the nitty-gritty and minute parts of this Zoom platform. All sorts of work, nitty-gritty and minute parts has been taken care of by efficient guidance of Mr. Amit Jain, Mr. Shonal Shah, Mr. Jignesh. We express our sincere thanks to all of you from core of our heart, and we expect your wholehearted cooperations in future program too. Now, we are very much thankful to silent background workers, and they have worked silently, and many of us may not know that they are working so hard in the background. They are none but they are Mrs. Shreya Chattopadhyay, Mrs. Ujjaini Sarkar, and Mr. Chandan De. They have worked hard in the background all the time without coming in the front side for last few weeks to make this conference possible. And last but most important section of a conference is none but the audience. Without their participations, without their interactions and attendance, no conference can be successful. So you will be very delighted, and you will be very much encouraged to know that more than 1,100 attendees logged in yesterday, and this 1,100 and this yesterday's coverage has been overwhelmed by today's logged in attendees. So we are really very much encouraged by your active participations to hear this sort of first ever World Congress on adult immunization. And we expect this sort of cooperation, this sort of attendance in your future program too. Thank you all once again from core of our heart, and hand over the podium to our chairperson, Sir Dr. Kole, for concluding our session of today's extravaganza of first ever conference on World Congress of Adult Immunization. Hand over to Dr. Kole, sir.
So all the persons who are behind this program, we have mentioned, and I would also just add Dr. Amitavo Nandi. Many a time at the late night I have disturbed him, and he said no, that he is ready to advise me, and Mr. Amit Jain, regarding he has always told that it is possible. You just go ahead, it is possible, and I have got his immense support. So whatever I have told, that the journey has started, and we need help from all of you, and definitely we will be able to do something to improve the present scenario of adult immunization in our country. Thank you very much.
Yeah. And we are really thankful to Dr. Amitavo Nandi sir, being so much aged. His brain is fully active, and he has shared his experience, his knowledge and everything. and we are having a good friend of us, that is Dr. Mohammad Saidur Rahman from Bangladesh. Thankful to you also, sir. Please join and share your experience in our future endeavor too. Thank you, all of you.